So good morning or good evening, depending on where you are. Welcome everybody to the 5th Fortaleza Austral Spring School. My name is Luisa Mello and I will be the moderator of our event. First, I would like to inform you that there is a real-time translation and the questions must be written in the chat. The school was organized in response to the need for a better understanding of the impact of the pollution in marine ecosystems and coastal communities. This free course will present a comprehensive overview of the problem and provide best practice and harmonized methodologies for developing research to quantify and assess the level of pollution as the response to incidents and disasters. The event is organized by the Postgraduate Program in Tropical Marine Sciences, together with the Institute of Marine Science of the Federal University of Ceará, LABOMAR in Brazil, and the Atlantic International Research Center, AIR Center. In addition, the event has the collaboration of other postgraduate programs. To our open ceremony, I would like to pass to the director of LABOMAR, the Professor Maria Osileia Bezerra Menezes, for her pronouncement.
Ela é muito especial. Além de uma programação fantástica com excelentes palestrantes, a 5ª Fortaleza Austral Spring School, Internacional Spring School Marine Science, ela é muito especial. Além de uma programação fantástica com excelentes palestrantes, comemoramos a assinatura de um importante memorando de entendimento que entre si celebram o Atlântico International Research Center, o ES Center, e a Universidade Federal do Ceará, através do LABOMAR, um acordo de cooperação técnica bilateral entre Brasil e Portugal, envolvendo países atlânticos, né? O Brasil ele tem uma ligação emocional com Portugal e racional com reconhecimento político do papel vital do oceano. Isso nos traz aqui hoje. É uma construção solidária entre instituições de pesquisas e entre os países que têm uma cultura comum. E para celebrar isto, eu gostaria de ler um trecho da Viagem do Descobrimento, de Eduardo Bueno, e é um trecho sobre um morro à final da Páscoa. Com tapetes flutuantes, elas surgem de repente, com muita tranquilidade, muita quantidade, balançando nas águas translúcidas de um mar que refletia as cores do entardecer. Os marujos as reconhecem de imediato, antes que sumissem no horizonte. Chamam-se botelhos, as grandes algas que dançavam nas ondulações formadas pelo avanço da frota imponente. Pouco mais tarde, mais ainda antes que a escuridão se estendesse sobre a amplitude do oceano, outra espécie de planta marinha iria lamber o casco das naves, alimentando a expectativa e desafiando os conhecimentos daqueles homens temerários o bastante para navegar por águas desconhecidas. Desta vez eram rabos de asnos, um emaranhado de ervas felpudas que nascem pelos penedos do mar. Para marinheiros experimentados, sua presença era sinal claro da proximidade de terra. Se ainda restassem dúvidas, elas acabariam no alvorecer do dia seguinte, quando os grasnados de aves marinhas romperão o silêncio dos mares e dos céus, as aves da anunciação, que voavam barulhentas por entre os mastros e velas, e que se chamavam furabuchos. Após quase um século de navegação atlântica, o surgimento desta gaivota era tido como indício de que, muito em breve, algum marinheiro de olhar aguçado haveria de gritar a frase mais aguardada pelos homens que se fazem ao mar: terra à vista! Bom, com essa exclamação cheia de alegria, terra à vista, eu digo, terra à vista, a ciência que nós precisamos para o oceano que nós queremos. Nosso futuro começa agora, descobrindo e avançando na ciência.
Thank you, Professor. And now I would like to invite Professor Augusto Teixeira de Albuquerque, Provost for International Affairs and Institutional Development of Federal University of Ceará.
Obrigado, Luísa. Eu gostaria de dar as boas-vindas a todos os participantes. É para a UFC uma satisfação enorme participar dessa iniciativa e, principalmente, observar a pujança do nosso Instituto LABOMAR, ao qual eu já gostaria de parabenizar pela realização, em nome dos professores Osileia, diretora do Instituto, o professor Luiz Ernesto, do Programa de Pós-Graduação, bem como o professor Tomaso, que é nosso professor visitante e que muito nos honra. Esse evento que conta com a participação significativa com representantes de quase todos os continentes, como disse o professor Moutinho, mais de 150 inscritos, se eu não me engano, ele é extremamente pertinente. E o seu tema, que é quase um alerta, é um chamado à sociedade a repensar o nosso uso dos recursos, quando ele diz que o nosso ecossistema marinho se encontra sob pressão. Então, é com esse olhar, com essa sensibilidade, com esse senso de urgência para esse tema tão importante que esse evento se inicia. Então, mais uma vez, sejam bem-vindos à UFC. A UFC se sente, ao mesmo tempo, honrada e orgulhosa do seu instituto, que em muito tem ajudado no desenvolvimento da ciência marinha no nosso estado e também com alta repercussão internacional. Muito obrigado e um excelente evento para todos.
Thank you, Professor. I would like to pass the word to the Chief Business and Network Developer for the Atlantic International Research Center, the AIR Center, which provide the technical and scientific support to our event, José Luiz Moutinho.
Olá, tudo bem? Vou falar aqui em português, porque nós temos tradução simultânea. Me acostumar um pouco a voltar a falar português, eu tenho viajado muito. Eu queria agradecer, antes de mais nada, a possibilidade de fazer esse evento convosco, agradecer ao professor Augusto, a Osileia, ao Tomaso e ao Laércio, e também a Catarina Duarte, do Air Center, que está por trás de tudo, e obrigado também, Luísa, por moderar essa sessão. É sempre difícil apresentar qualquer coisa depois da Osileia. Todas as vezes que a Osileia faz uma apresentação e intervém, temos sempre poesia, temos palavras bonitas. A Osileia tem um conhecimento enciclopédico de livros e é difícil competir, mas eu só queria agradecer mais uma vez a possibilidade de assinar esse acordo com o LABOMAR, com o UFC. É uma relação que já começou há muito tempo, né, e agora ficou mesmo, digamos, assinado, formalizado essa relação. E eu queria só acrescentar algo mais. Professor Augusto, nós temos nesse momento 1.100 pessoas inscritas e 228 pessoas participando. Desses inscritos, vêm de 53 países diferentes. Então, é massivo, nós temos agora vários assistindo através do YouTube, e eu tenho certeza, ao longo do dia, as pessoas vão também participar e vão, ao longo do dia, encher ainda mais essa sala, que já tem mais de 200 participantes no momento. Então, é um enorme prazer poder partilhar com toda essa comunidade, o conhecimento, né, gerado ou associado ao LABOMAR. É um enorme, digamos, prazer mesmo de poder falar convosco de uma maneira, como é que eu posso acabar? De uma maneira muito objetiva de que o AirCenter vai apoiar em LABOMAR em tudo que é possível, né? Então, eu gostaria novamente de pedir, né, de agradecer todo esse apoio, de insistir, né, que continue essa relação depois da assinatura formal. E agora acho que passo de volta a palavra para a Luísa, porque eu já falei demais.
Thank you. Next, I would like to invite Professor Luis Ernesto Arruda Bezerra of the Coordination of the Postgraduate Program in Tropical Marine Science to deliver a welcome speech.
Graduação em Ciências. Bom dia a todos e a todas. Meu nome é Luiz Ernesto, sou docente do Instituto de Ciências do Mar, da UFC, o LABOMAR, e vice-coordenador do Programa de Pós-Graduação em Ciências Marinhas Tropicais. Estou aqui para dar as boas-vindas a todos e todas participantes da 5ª Escola Internacional do Programa de Pós-Graduação em Ciências Marinhas Tropicais. Após 5 anos, após 5 edições, podemos dizer que a nossa escola internacional se consolidou. E como nos anos anteriores, trazemos uma programação que conta com a presença de pesquisadores renomados que atuam na fronteira do conhecimento de vários países para discutir alguns problemas que afligem os nossos mares e oceanos. No caso, a poluição por plástico e a poluição por óleo, né? Eu gostaria de ressaltar que essa escola ela é totalmente gratuita, organizada por professores do programa de pós-graduação, que visa dar uma melhor formação aos nossos estudantes. Claro que essa escola já rompeu fronteiras, como é o interesse por ser uma escola internacional. E como nos anos anteriores, nós contamos com a participação de pessoas de diferentes países. Esse ano, contamos com inscritos de 47 países, o que faz a nossa escola internacional de fato. Após 2 anos de pandemia, onde a nossa escola aconteceu de forma remota, esse ano estamos fazendo uma escola de forma híbrida, que permite a participação de pessoas do mundo inteiro, mas também conta com atividades práticas realizadas presencialmente aqui nos laboratórios do LABOMAR. Também convidamos pesquisadores renomados que atuam na fronteira do conhecimento das áreas que serão discutidas aqui na nossa escola. Nesse sentido, eu já queria deixar aqui também, de antemão, um agradecimento à comissão organizadora, na figura dos professores Tomás Jarrizio, Laércio Lopes, Rivelino Cavalcante e Fábio Matos, que é o coordenador do programa de pós-graduação. Essa equipe trabalhou muito, com muito carinho, para trazer uma escola internacional de alto nível, do jeito que vocês merecem. Então, espero que todos tenham um bom aprendizado e uma boa escola internacional. Muito obrigado.
Thank you. And last but not least, I would like to invite Dr. Tommaso Giarizzo, visiting professor at LABOMAR, chair of the 5th Fortaleza Austral Spring School, for his welcome speech.
Good morning, good afternoon, and good evening to everyone, wherever you are. Thanks very much for joining us in the 5th edition of the Fortaleza Austral Spring School. I'm very happy and honored to have the opportunity to be the chair of this fantastic event. First of all, I would like to thank the director of LABOMAR, Professor Maria Auxileia Bezerra Menezes, and the coordination of the postgraduate program in tropical marine science for inviting me to assume this important role in leading this event that could not have been carried out without the partnership with the AirCenter and the great collaboration of the entire organizing committee, in particular, my colleague, Dr. Laércio Lopes Martins. The title of the 5th edition of the Austral Spring School is Marine Ecosystem Under Pressure, Current Issues and Future Challenges in Pollution Science. Before starting the day's presentation, I would like to introduce for all of you the relevance of the topic dealt in this event. Everyone knows that we live on a blue planet with the ocean and seas covering over 70% of the Earth's surface. But yet a lot of people don't know that a prime condition of human right is to live in clean, healthy, and sustainable marine and estuarine environments. So now is a good opportunity to remember 5 main aspects that prove how essential is a healthy ocean for all of us. The first aspect is the ocean is our life support system. 1/5 of the total human population, nearly 2.4 billion people, live within 100 kilometers of an oceanic coast, and most of the world's megacities are located in coastal areas. A second aspect is that the ocean provides food, jobs, and livelihood. All the marine and estuary ecosystem feed us through fishing and aquaculture and provide medicine, minerals, and renewable energy resources that sustain the world's 7th largest economy, with an annual economic value of more than $2.5 trillion. Besides, the oceans links more than 180 coastal countries and provide through maritime transport the movement of people and goods that represent over 80% of the volume of international trades. A third important aspect is the ocean is the Earth's natural climate moderator, regulating climate and influencing weather patterns around the globe and affecting everyone else. The ocean also produces around half of the oxygen that we breathe, and much of the carbon dioxide emitted by the human activity into the atmosphere is absorbed by our seas, which help to fight the climate crisis, trapping this warming gas and acting as a carbon sink. The fourth aspect is the ocean owns amazing wildlife that is essential for the structure and functioning of marine ecosystem, allowing them to be productive, resilient, and adaptable to environmental change on local, regional, and global scale. At the present, there are more than 241,000 accepted marine species, from tiny plankton and bacteria to huge whales and giant kelp. But unfortunately, this is on the tip of the iceberg, since around 80% of the ocean remains undestroyed, and 91% of marine species remain undescribed. Finally, the ocean has a fundamental role to play for all culture, providing spiritual well-being to humanity. Beach is one of the best places to alleviate stress, and many of us enjoy beaches and coastal area for swimming, diving, surfing, sailing, or simply stay close to the water, increasing the spiritual connection to the ocean. So I hope that it is evident to all of you that we are part of the ocean. We rely on it to provide us with essential and useful service for our lives. But in contrast with the irresponsible behavior, we also use the planet as a sink of our waste products, such as carbon dioxide, plastic, and other forms of waste, including oil pollution. Among the numerous and most diverse environmental stressors, this year we will focus the school on 2 very worrying topics: plastic and oil pollution. On the first day, we will have a fascinating lecture given by international experts, whom I would like to thank for having accepted to participate and contribute to the success of this event. In the first session, with the objective of showing and contextualizing the problem of plastic, colleagues will present important findings on plastic thread, polymer composition, and plastic ingestion. In the second session of the day, they will focus on the methods to be adopted to study the problem of plastic pollution, and therefore participants will be able to learn about some of the most appropriate tools to extract plastic from different metrics, such as sediment, water, and biota, as well as the potential use of drones for assessment and monitoring the solid waste. So welcome on board to the 5th edition of Fortaleza Austral Spring School. Thank you for staying with us.
Thank you, Professor Tomás Gierizo. This is the end of the opening ceremony. Now we start our first session, theme Fate and Impact of Plastics in Marine Ecosystems, with Dr. Raymond Ward as chair of the session, a visiting professor at Colégio de Estudos Avançados at Federal University of Ceará and professor at the University of Brinton. And the speakers, Dr. Sedat Gundongo, Dr. Colin Ward, and Dr. Robson Santos. There will be all three speakers' presentations and then a 20-minute roundtable where the questions will be answered by the speakers. The session will begin with Dr. Sedat Gundongo presentation entitled Plastic Waste Trade from Global North to Global South as Source of Marine Plastic Pollution. Sedat Gundongo is a marine biologist, work on marine plastic pollution at Faculty of Fisheries, Çukurova University in Turkey. In his works, he draws upon an array of disciplines such as marine biology, chemistry, agricultural science, governance, stakeholder involvement, communication, perception, and knowledge about different regulatory bodies. So welcome, Professor Sedat Gundongo. The screen is yours.
Thank you, Lisa, for this good introduction. Let me share my screen. Hello, everyone. It's really good to be here with you all. First of all, I would like to express my special thanks to the organizer for inviting me to this wonderful event. Today, I will try to present the connection between the marine pollution, marine plastic pollution, and the plastic waste trade, which is a kind of secret connection of this issue. The first, here's the outline of the my speech and my talk. I will first give a brief info of the background of the issue and then connection with waste and the waste trade and what's waste, the meaning of the waste and its connection with colonialism and global pollution and dirty waste, the dirty waste trade, clean west and dirty east, and kind of default quotation happening regarding with plastic waste trade, and then give some conclusive remark about the issue. So first, we should define what's waste. The concept of the waste is intensely familiar to us. It's something we meet every day. Although to define exactly what makes something waste is far from obvious. Waste may be the only thing that no one owns, but that everyone generates. So currently, waste is considered as a raw material in a circular economical mentality. So when you define waste as a raw material, it can be trading activity topic. So this is the point, one of the points that is behind and the background of the waste trade. So the good or the bad, that is the question. If you define waste as a good, but for whom? And if you define plastic waste as bad, but still you should give answer of for whom. So in general, avoiding a potentially toxic material is the more important thing behind the waste trade. This is the exact reason why this plastic waste moving transboundary limits. So if you say waste is good, so then can you send this waste to another part of the world? Yes or no? It depends your position on this issue. Or if you say waste is bad, then what will you do with this waste? Will you dispose in your own territory, or still you have chance to send this waste to the other countries? So the behind of this issue is kind of the root of colonialism. There is a two kind of colonialism. One is the old style colonialism, which is not exist anymore. But second, the new understanding, new meaning of the colonialism is almost equal with waste colonialism. We call it toxic waste colonialism. So the most important evidence that waste trade has a colonial reflex is in an article published in 1992 by a newspaper called Jornal do Brasil. The event, called the Summers Memo, belongs to an internal correspondence from the World Bank. In this correspondence, Lawrence Summers, which you can see in the slide, the chief economist of the World Bank at that time, sarcastically tell us that sending waste to third world countries was a form of trade they desired, explaining in a colonial tone that the environment of the developed countries should be clean and good. Summers apparently thinks that it's not necessary for the less developed countries to live in good environment. So he says the direction of the waste trade actually has a clear character, is colonialist character. So the main theory is disposal cost of toxic waste in the rich countries has high cost, and sending this toxic waste to the poor countries has less cost. So disposing the waste which is generated in generated country has to need more cost. So sending this waste to the low developed countries is cheaper. This is the main theory of waste shipment. So then we should look the general trend of the plastic waste movement in the global scale. So it's obvious that we can see all this waste flowing from global north to the global south countries. The number and the amount, the volume of the exported waste decreased year by year, but still we have almost 4 million tons of plastic waste every year flowing from high developed countries to the low developed countries. So the main destinations you can see in the second graph in 2021 to Turkey, one of the first destination, and the other non-OECD countries, mostly located in Southern Asian country territory. So here you can see the movement of the waste from the developed countries to the Asian countries. Mostly it ends up in Hong Kong, India, Malaysia, Indonesia, and Vietnam. So all this United Kingdom, European countries, and United States and Japan sending all the waste that they generated to those countries. So what's happening after that movement? You know, this importer side flowing more and more plastic to the ocean. You can see this graph that the importer side has mostly landfilling and open dump practices in terms of waste management. So we can say that global plastic waste export hindering local waste management. So almost all of the importers have a big landfilling and collection problem. So this makes the importer bigger polluters. So, but in theory, but in reality, all this, most of this waste flowing into the ocean and the coastal side in the importer part belongs to exporter countries, but this is hidden part of the issue. So you can let's close look at Malaysia, Vietnam, and Turkey's waste management practices, and you can see that all these three countries, which are the main destination of waste, they have similar pattern of the plastic waste management. So all of them has high amount of waste that they generated, and also they are importing plastic waste from those countries, the exporters, and they even cannot manage their own waste, and they are continuing to import, and the exporter side still flowing their waste to those countries, even they don't have enough capacity to manage. So this picture generates a new motto, like a clean west and dirty east. So you can check this two picture. One is the winners of the recycling, and the second one is mismanaged plastic waste. You can see this side of bottle includes only the countries in the southern Asia and some African countries, and also Turkey. So in the winning recycling table, you can see that main exporter, they are really good for collection, sorting, and storing their plastic waste. And also they are very good to incineration. So they incinerate mostly 50%. For instance, for South Korea, is 90% of their waste they are incinerated. But on the other hand, the remaining part of the waste that they generated sending to those countries in this side of bottle. So what should we say? The countries in exporting side are clean and more responsible for environment, and the importer part are not responsible when we compare with this exporter countries. So that's, of course, depends on your position when you give answer this question. So the second issue with the waste trade, the microplastic footprint, which you probably know, microplastics are plastic particles smaller than 5 millimeters. So the mechanical recycling, one of the main activities regarding plastic waste, import export, because most of the importers are processing plastics that they import with mechanical way. So the mechanical recycling is one of the main source of microplastic pollution in the waterways. Like I would like to show you this video. This is from a wastewater treatment plant in Turkey, which receive huge amount of wastewater from these recyclers that import waste from other countries. You can see this is all microplastics. Nothing to do this because there is no way to dispose all this waste. Only they should incinerate, which is not really a desired way of disposal of such kind of waste. And they are just storing in the behind of the facility and then sending them to the official landfill. So this is the way of the disposing imported plastic waste in imported side. So what's happening after that? You see the Turkey as an example. Turkey is one of the country in Mediterranean region that has most polluted coastlines. You see all the color are red. I mean the highest color, but for Turkey is getting more red. There is no enough pattern of red, and then they turn into purple. So per kilometer and per day, 31 kilogram of plastics ends up in the coastal part of the northeastern Mediterranean coast of Turkey. So we can say that the Turkish coast is one of the most polluted coasts in the Mediterranean Sea, and the previous slide that I showed you was such amount of microplastic waste ends up in the Mediterranean Sea via Seyhan and Ceyhan River, which is two most polluted river in Mediterranean Sea. So what should we say that the Turkey is one of the biggest polluter in the Mediterranean Sea, or the exporters feeding this pollution activity by sending the waste to Turkey? So we can say the second option for this. So in general, you can see this map that most of the importers and other less developed countries are responsible for plastic waste in the ocean. You see this main exporter has quite yellow one, not the dark yellow or orange, because all this mapping activities, all this statistical interpretation has also colonialist background. So this is the conclusion. This picture from a recycling facility located in Turkey, receiving plastic waste from UK and European Union countries. So they are just the workers are working in very bad conditions, and all these facilities are mechanical recycling facilities generating billions and billions of microplastics, and there is no effective wastewater treatment activity. So still, they are generating microplastic because of this import-export issue. Also, because of this waste import hindering waste management infrastructure of the country, so the collection rate of Turkey is much more than 10% of the total waste that we generated. So there is a tremendous impact of plastic pollution in the global south, and the pressures southern nations are under to continue to import plastic products and to manufacture plastic domestically. So under the status quo, plastic pollution is waste colonialism in the global south. Plastic production, manufacture, and the pollution benefits the economies of the global north while devastating the environment, livelihoods, health, and cultures of the global south. The plastic waste exported to the global south contaminates the global south's food and water sources, ancestral and cosmological connections, and its people across generations. Plastic pollution is an ongoing colonial violence on the land, ocean, air, and all kind of environmental compartments of the global south. Thank you.
Thank you very much, Dr. Sadat Kundongo. I hope you all enjoyed this amazing presentation. Now I will introduce the second speaker, Professor Colin Ward, who will give the talk entitled Insights into the Perceived Persistence and Photochemical Fate of Plastics in the Ocean. Professor Colin Ward is Associate Science in the Department of Marine Chemistry and Geochemistry at the Woods Hole Oceanography Institution in Massachusetts, USA. He earned a BS and MS in Environmental Science from the Ohio State University and a PhD in Earth and Environmental Sciences. His research characterizes how fast sunlight and microbes alter the physical and chemical properties of organic carbon in aquatic ecosystems. He works on a wide range of organic carbon types, including natural organic matter, crude oil, and plastics. So welcome, Professor Colin Ward. The screen is yours.
Thank you so much. Can you all hear me? I assume so.
Yes, yes.
Great. Thanks for the introduction, Luisa. Let me share my screen real quick. Great. And are you looking at the actual presentation? So,
no, we've got the
presenter view.
The presenter's view, yeah.
Okay, you should be better now, right?
Yeah.
Yeah, all good.
Great, thank you so much. Sorry, I wish I could have attended the practice session, but the sun had not risen yet. So bear with me, it's a little early here. I'm so honored to be here. Thank you so much for everyone for the invitation. Thank you to all the speakers. It's humbling to see the list of speakers that are in the session. It's really a great honor to be here and talk to you about the work we've been conducting in my lab over the past few years. My talk is really focused on this concept of the persistence of plastics in the environment. And we're going to shift gears and think about what happens to plastics in the ocean and in surface waters. And we're really honed in on this concept of persistence because it is a key regulatory sort of term or framework. And the general understanding of this property is that the longer pollutants last in the environment, the more harm they can pose to aquatic and ecosystem health. And the challenge we're having is that, as I'll show you, is that there's not great understanding of the actual persistence of these materials in the environment. And we're trying to work on understanding or better understanding how these materials react in the environment and how long they last in the environment. The motivation comes from this concept that we're all aware that we're using more and more plastic every year. The demand for plastic is climbing exponentially over time, and it has been since its inception in the 1950s. And with this should come, a rise in the amount of plastics that we're detecting in the environment. But there's lots of stories out there demonstrating that perhaps there is not this this rise in the amount of plastics that we find in coastal waters. So this is a report from Francois Galgani's study in 2021, where they simply state that while the predicted total global amount of plastic litter is increasing, as suggested by models, the parent steady state situation of plastic quantities observed in coastal systems challenges our ability to predict plastic sources and fate. And this essentially suggests that perhaps these materials are breaking down on timescales that are relevant for their input into these coastal systems, creating this steady state scenario. The challenge we're having is we actually don't have a firm grasp of how long these materials last. And when I started thinking about this problem, I started looking at a lot of the sort of infographics that were floating around, right? I think we're all familiar with these. We've seen these infographics posted in, on beaches, in different, on the internet. We see them in textbooks. They're basically just graphics that describe how long different types of materials last in the environment. So I did this really in-depth review of these different infographics. In total, we reviewed 57 graphics and documents. There are 255 estimates that we compiled. They were sourced globally, 3 different languages, 13 countries across 4 continents. And these graphics were prepared by all sorts of organizations, from the government to nonprofit organizations to the media, lesson plans, academic textbooks. So this one on the right here, this is a textbook for environmental geology. And it's a textbook that I actually used to teach environmental geology at the University of Michigan about a decade ago. And what we did is we summarized all these estimates for how long different articles last, and we created one figure. And this figure was sort of the synthesis figure that formed our article. And in this figure, what you're seeing is all the estimates compiled and summarized as a function of article type on the y-axis. And then the x-axis is the lifetime estimates, right? The black bars represent the range in the estimates, and the red dots represent the means. And I want to take you through sort of 4 key take-home messages that we had from this exercise. The first is that you can see the black bars are extremely wide. And what this tells us is that we have very low confidence in how long these materials last in the environment. In some cases, they range from 1 all the way out to forever. And the best example I can give, something that hits home, is the lifetime for a plastic bag shown here. So I am currently, this is North America, I'm currently in Massachusetts. And if you zoom in, I'm sitting right now in Woods Hole, Massachusetts, which is on Cape Cod. Boston, Massachusetts is to the north, and Providence, Rhode Island is to the northwest. And Nantucket is about 65 kilometers across Martha's Vineyard Sound to the southeast of us. And the question is, well, how long does a plastic bag last? Does it last 10 years or 1,000 years? And if you ask the federal government, this is the United States National Oceanic Atmospheric Administration, they suggest that a plastic bag lasts 1 to 20 years. But if you ask the local government in Nantucket, which is 65 kilometers to the southeast of the Woods Hole Sea Grant estimate that I showed you on the last slide, they suggest that a bag lasts about 500 years. So we're 65 kilometers apart, yet 25 to 500-fold differences in the lifetime of the same exact plastic good. And I would state that if our goal is to determine the risks of these emerging pollutants and write relevant, meaningful policy, I think we need to do a better job of understanding how long these materials actually last in the environment and how they behave. The second take-home message is that none of these 255 estimates are actually rooted in peer-reviewed science. If you dig deep and you try to find out where the estimates came from, it seems like they all originated from 3 different sources. The first is NOAA, the Sea Grant programs and the marine debris programs. The second is the Mote Marine Lab and Aquarium in Sarasota, Florida. And the third is the South Carolina Department of Health and Environmental Control. I talked to multiple librarians at different locations over time, and essentially they stated that most of these estimates have originated from Mote Marine Lab and NOAA, and then they were not rooted in peer-reviewed science. And that's a problem because peer-reviewed science should be informing policy, right? The third take-home message is that unlike the recycling of plastic goods by humans, we appear to be really, really efficient at recycling these non-peer-reviewed lifetime estimates. And the best example is the lifetime estimate for fishing line. So there were 37 different estimates for fishing line, and they all suggested that the lifetime of fishing line in the ocean is 600 years. It's really unlikely that there's 37 independent peer-reviewed studies out there that are reporting this lifetime of exactly 600 years. And really what's happening is folks are seeing one lifetime estimate and just taking that number and reusing it over and over and over again without scrutinizing the science or lack thereof behind the estimate. And the last take-home message I want to share with you is that generally these graphics lack precision in the wording, right? There's no definition of what degradation is. And that's a challenge because degradation into carbon dioxide or into transformation products that dissolve into the water or to smaller microplastics, they all have, and they all pose different risks to ecosystem health. So we need to understand, whether or not, or which pathways of degradation are occurring. And another example is there's no environmental context. So generally speaking, we're not talking about where the degradation is happening. And that's a challenge because we would expect the lifetime of these goods to be a lot different in soils versus rivers versus lakes versus the open ocean versus sediments, right? So we need to have more nuance about the location and the pathways of degradation that are occurring. So I quickly want to sum this up before I transition into more nuanced science that my lab is conducting to tackle some of these knowledge gaps. I just want to address what we learned here and where we're going now. The first, again, is that there's lots of uncertainty in these lifetimes. The estimates are not peer-reviewed, so we definitely need more peer-reviewed knowledge about how long these materials last. It appears that we've been carelessly recycling these estimates over and over again. And then lastly, there's poor precision with the wording. We need to be more precise and think about the pathways of degradation and where degradation occurs. Before I move on, I want to explain or make clear that this review was not written to justify littering. It was more written to bring everyone on the same page about our knowledge surrounding this important topic, and then make clear that there is an opportunity for scientists to do research into filling these knowledge gaps about the persistence of these materials. And also, I want to state that stakeholders really want this information. We wouldn't have 57 infographics on 4 continents if there wasn't a demand for this information. So it's really pertinent and pressing that we move forward with this type of research. And that's exactly what my lab's been doing over the past few years, is thinking about how we can design experiments to try to put estimates for the lifetime of these materials. Before I dive into some key findings, I just want to share with you sort of the old view, in my perception, the old view of how plastics behave in the environment. And in particular, I want to highlight that I'm only focusing on photochemical degradation. This is degradation by sunlight. There's other forms of degradation, but I don't have time to go into that now. So traditionally, if you look in the literature over the past few decades, there is generally this perception that plastic fragments will just break down into smaller and smaller pieces. So sunlight is breaking these materials, large pieces of plastic into smaller pieces of plastic. So macroplastics into microplastics. And the concept is that there's no changes to the chemical properties. It's only changes to the structural properties. So these larger items are going into smaller items, but there's no changes to the polymer itself. This was supported by a statement by the GCMT report in 2019, which is only 4 years ago. So as of 4 years ago, the joint group of experts, held this assumption about the fate of plastics in the sunlit sea surface. And a couple of years ago, we, and along with many, many other folks in the community, started discovering that these plastics are actually very reactive in nature, right? Especially when in the presence of sunlight. And what happens is sunlight certainly breaks up these plastics into smaller and smaller pieces, but they also break these plastics down into water-soluble chemicals, right? So these are transformation products that dissolve into water, as well as gases that can get released into the atmosphere. And we wrote a paper in 2019 that described this for polystyrene. Many, many other studies, dozens and dozens of studies have also written papers like this since. And since then, we've really been exploring, how this experimental framework, or how this framework of the fate of plastics in the environment applies to different types of plastics. And we've really been embracing this concept that plastics are a complex contaminant class. So this is a schematic that I'm borrowing from Chelsea Rockman's 2019 paper, where essentially they just argue that we need to think beyond, the core polymer types of plastics, and instead think about the formulations of plastics, the additives that are in the plastics, the types of uses that we have, the sizes, the morphologies, the colors. All of these could potentially dictate or control how long these materials last in the environment. So I'll give you one example of how we're embracing this, complex contaminant class perspective. We first started studying the degradation of plastic shopping bags. So these are these single-use shopping bags that are banned in about half of the states in America. And what we did is we collected bags from major retailers. So we were using CVS, Target, and Walmart. These are some of the major retailers in America that use these bags and give these bags out with their products. And we're comparing the reactivity of these materials to a control, a polyethylene, pure polyethylene standard that we bought from a manufacturer that does not have any additives in it. And one thing we found is that no matter what type of material we used, when we put it in water, we always found that a little bit of dissolved organic carbon would leach out in the dark. But then when we shine light on that same plastic, 4 to 5-fold greater dissolved organic carbon would leach out in the presence of light. And what's happening here is sunlight is breaking up the polymer backbone and oxidizing it, and small chemical fragments are leaching out and are dissolving into the water. The next question we had is, well, what's the chemical composition of all these different organics that are leaching out in the dark and in the light? So what we did is we concentrated the dissolved organics, we removed the salts from them, and then we analyzed them using a high-resolution mass spectrometer at the National High-Field Magnetic Laboratory at Florida State University. And what I'm showing you here is the raw mass chromatogram of the analysis. On the top is the dark controls in black, and on the bottom are the light exposed samples in orange. And we're going from 400 Daltons on the x-axis to 1,000 Daltons. And essentially what I want you to see is that there's just 10-fold greater, more peaks that are popping up in the light exposed treatment compared to the dark control treatment. So not only are we creating, more dissolved organic carbon that dissolves into the water, but the complexity is about 10-fold higher. So in the dark, there's about 200 peaks that are unique to the dark. And you can see when we zoom in, you see that there's only about 4 peaks from a mass range of about 570 to 600, whereas there's hundreds of peaks that are in the light exposed treatment. And totally in the light exposed treatment, there's over 13,000 unique peaks that are assigned. One of the next questions we had was, well, how does the formulation of the plastic relate to the composition of these different molecules that are being generated by sunlight? So we started sort of analyzing what the formulation was of these plastic bags. And we really honed in on the inorganic aspects of the plastic, right? So in this case, what we find is that the pure polyethylene has no inorganic carbon. All of it is organic carbon, whereas up to about 35% of the consumer plastics is actually calcite or calcium carbonate, which is used as a filler to bulk up the plastic and to make it cheaper. But in every single consumer plastic, we also detected titanium dioxide, which is often used as a pigment to make it opaque. And that titanium dioxide is also a photocatalyst, which can impact sort of the chemical composition of the dissolved organic products that are being generated when we shine light on them. And that's shown really clearly here. This is what we call a Van Crevelen Van Crevelen diagram, where every single point corresponds to a molecule detected using high resolution mass spectrometry. And it's plotted according to the oxygen to carbon ratio on the x-axis and the hydrogen to carbon ratio on the y-axis. And really what I want you to see is that the blue dots, which were produced in the pure polyethylene, separate very nicely from the red dots, which were unique to the consumer plastics. So this tells us that these additives are likely driving both the rates of degradation as well as the composition of the products that are produced when exposed to sunlight. Another example of embracing this, complex contaminant class perspective is thinking about how molecular weight plays a role. So in addition to the types of plastics, we're also now thinking about how the molecular weight of those plastic change impacts its fate. And to do this, we developed a method called diffusion order spectroscopy. It's an NMR-based method. It's high throughput and it's precise for the screening of polymer molecular weight. Essentially, small molecules diffuse quickly, so a lot of the resonance signal is attenuated, and then large molecules diffuse slowly, so most of the resonance signal is retained. We applied this method to polystyrene, and the first thing we did was just to get a sense of, well, what's the range of molecular weights for polystyrene? How much does it range? And it turns out that the range is over a factor of 2. So when you buy polystyrene at the store, the molecular weight can vary from anywhere from 100 Daltons to over 200 Daltons. Kilodaltons, sorry. And the mass or the average molecular weight really depends on the stiffness of the material or how hard it is. Basically, things that are harder or film-like materials have lower molecular weight, or have higher molecular weight compared to foams, especially soft foams like packing peanuts. Our next question was to try to link these differences in molecular weight. So we grabbed representative samples from these different materials and we shined UV light on them for increasing amounts of time from 0 days all the way to 10 days using a UV LED array. And then we just tracked the decrease in molecular weight over time. And for all samples, molecular weight decreased, but by far the biggest decrease was for the low molecular weight samples, the foam samples, relative to the coffee lid or the hard film samples. Then we wanted to know, well, is this chain scission of the polymer backbone impacting the bioavailability of these materials or the fate of these materials in the ocean? So what we did is we actually fed the day 0 samples and the day 10 samples to native microbes in the ocean, and we measured the light minus dark respiration, right? So this is the stimulation in respiration due to sunlight exposure or due to UV exposure. And in all cases, we had a stimulative effect. What this tells us is that sunlight is breaking the chemical backbone of polystyrene into smaller and smaller pieces and making it more bioavailable for the community. And this is happening on timescales that are highly relevant for transit in the ocean, suggesting that this is an important sink of these materials in the surface ocean. Okay, so I'll wrap up with a few conclusions. The first is generally we have a poor understanding of how long these materials last in the environment and what they break down into, and what the controls are of this process. The second is that fragmentation is far more complicated than large pieces of plastic going to small pieces of plastic. There's chemical chain scission and oxidation that occurs that can transform the plastics into water-soluble compounds and gases. The third is that sunlight exposure appears to be a governing control of the persistence of plastics in the ocean. So I think a lot more research needs to be done on thinking about, well, what factors matter most? What factors are driving photooxidation and the rates of photooxidation as well as the composition of the products? The fourth is that plastic formulation, including both the additives and the molecular weight, are controlling the rates and the pathways of degradation. So there's a lot more work that can be done in this area, especially when thinking about how to leverage this information into new plastics that degrade faster. And then lastly, I think it's essential that risk assessment and fate models account for these light-driven transformation processes. You know, these processes are happening on timescales that are relevant for exposure to aquatic animals and for the fate of these materials in the ocean. So we need to start accounting for them in our models. I'll thank everyone for organizing this really exciting school or online school, and also to funders for supporting all of our research, and to all of my colleagues for helping out with doing this research. Thanks.
Thank you, Professor Colley-Ward. It was a very interesting talk. The last speaker of the first session is Professor Robson Guimarães dos Santos. He is an associate professor at the Instituto de Ciências Biológicas e da Saúde at Federal University of Alagoas in Brazil. His research focuses on plastic pollution and sea turtles biology. In the last couple of years, he has spent more efforts on the understanding of the causes and consequences of plastic ingestion by wildlife. So his presentation is entitled Plastic Ingestion as an Evolutionary Trap. The screen is yours, Robson.
Thanks, Luisa, for the introduction. Let me share my screen right now. And I think everything is all right, right?
Yeah, we can see.
Okay. First, thanks for the invitation for this fantastic event. I'm really enjoyed to be a part of that. And like Luisa said, I'm Robson, an associate professor at the University Federal de Alagoas, Brazil. And today I will speak about plastic ingestion as an evolutionary trap, and I will try to address the fundamental question of why animals ingest plastic. So, spoiler alert, it will be a lot of theoretical stuff, so be prepared. So first, we need to have in mind that we are in the middle of an environmental crisis, right? And today I will speak about plastic ingestion by wildlife, which may be understood as intersection between these two planetary boundaries here. One related to biodiversity crisis, another one to the pollution in general, where plastic is the key role of this boundary. And it's through these lenses that we live in a world where the co-occurrence of multiple anthropogenic stressors is the norm, that we should think about plastic pollution and its impacts, right? So let's dig in the topic. Please take a look at this image and what do these species have in common, right? You can think about taxonomical relationships or ecological traits. At the first glance, the species showed here may have little in common, but one thing connects them all. All of these species have been ingesting plastics. And today, over 1,500 species have been reported ingesting plastics, from lands to sea, right? And several branches of the tree of life are now contaminated by plastics. It's not something just a problem for one taxonomical group. It's widespread in the tree of life. And we can also see the plastic pollution using an ecological perspective. And you see that animals from all trophic levels are ingesting plastics. Here, animals represented in blue. Animals in blue are that we have reported ingesting plastics. But if you want to take a look in the food web as a whole, you will find that most of the nodes of the aquatic food webs are contaminated as well. And you can try other points of view, and we'll find similar results. For example, when you look at the catch of marine species for human consumption, we found that 9 out of the 10 most consumed fishes are reported ingesting plastics. You can look to the red list. When you evaluate the IUCN red list, we'll find species ingesting plastics in the most of red list categories. And we know that plastic ingestion can affect animals by a diverse set of mechanisms, affecting health, growth, reproduction, and may ultimately leads to population decline. The negative effects of plastic ingestion are extensively reported for several species, ranging from little to chronic effects. And the chronic effects are the more common ones. And potential effects on the population level are just beginning to emerge, because most of the studies today are in the individual level or levels or small levels than biological organization. So, why animals are ingesting plastics? When you think about this question, probably this kind of image comes to mind, right? Plastic bags and jellyfish. But this idea of close resemblance between plastic and a food item is only part of this story. But before answering why animals are ingesting plastics, it's important to understand one key concept, the concept of evolutionary trap. And we need to recognize that the animal decision-making process is not perfect, right? And that we live in a rapidly changing world, which makes everything more difficult to make a good choice. Human activities are changing our environment, creating a great number of low-profit options that mimic cues of high-profit ones, which draw animals into evolutionary traps, as they are misled into make bad choice. Here we have two of many examples of evolutionary traps. The first one is related to identify cues from a food item. For example, this little frog here was trying to eat that insect, but that's what happened. And the second one is to identify cues from a mate. And the beetle is trying to copulate with this bottle. So we have original environment, original cue, and original response, leading to an expected outcome. For example, ingesting a food item, or copulate with your female. And now we have human activities changing the environment. We have a new environment. And we have addition of new environment, new cues that mimic original cue. And we have now our original response, but at this time, to a new coup that leads all animals to evolutionary trap, to bad choice, deleterious behavior response. In this sense, plastic pollution can be considered a evolutionary trap, perhaps the most ubiquitous one, because the sudden appearance of plastics in the environment, and when I talk about sudden appearance, you need to have in mind the evolutionary time, where all the choices, the decision rules for choosing for item or another maybe seen in evolutionary time, right? Creates several, this sudden appearance of plastics creates several low profit options that mimic cues from a food item, leading to plastic ingestion, which is a deleterious feeding response. And before appearance of plastics in the environment, the low profit options was much less common. If you think about aquatic animals, for example, we can think that the animal low profit option will be like a piece of wood floating in the water, or a feather that have no nutritional value. And ingesting these items were probably harmless, but this kind of item was very uncommon in the environment too. And recognizing that plastic ingestion is an evolutionary trap, it's important to give us theoretical tools to address the fundamental question of why animals are ingesting plastics. And this question was the subject of our review on science published last year, when we unify evolutionary, ecological, and cognitive approaches under the evolutionary trap theory to identify the main factors that may drive plastic ingestion. So back to our question, why animals are ingesting plastics? First, to be ingested, a plastic don't need to perfectly mimic a food item. It just needs to cross an acceptance threshold of a given animal. And we'll come back to this point later, of the acceptance threshold. And in our study, we identified 4 main drivers for plastic ingestion, all of them supported by the literature. And the first one's plastic availability. And we can think as the amount of plastic in the environment, which will influence the plastic encounter rate, the encounter rate. In other words, the chance of an animal encountering a piece of plastic when it's foraging. I think this is probably the easiest driver to understand, but we have 3 other drivers that are a little bit more complex and demand a little bit more theory. And the first of them, it's level of prey resemblance to plastics, the case of jellyfish and plastic bags. But the resemblance could be physical or chemical, for example. The food selectivity, and when I say food selectivity, you may think about the generalist to specialist range of feeding behavior, and the nutritional state of the animal. So it's more restricted to the individual, a factor restricted to the individual, not the species as a whole, where the risk of starvation will play an important role in ingesting plastics. So to understand plastic ingestion, first, we need to be humbler, to set us free from our sensorial cage in order to broaden our perception of what may be considered edible for different species. Second important thing, we need to recognize that sensorial systems are imperfect. Therefore, they don't give a complete understanding of the world. Our sensorial system included in this. And evolutionary history highlights that species are constantly subject to a trade-off between 2 types of errors: accepting a low profit option or rejecting high profit ones. The decision-making rules of the animals will be in terms of these 2 types of errors. So that is where the signal detection theory steps up to help us understand the plastic ingestion. So let's think about this. In the real world, it's not just a matter of detecting the signal emitted by a high profit option, right? Because there is a lot of noise in the world. So we need to tell apart the noise emitted by a low profit option from the signal emitted by the high profit one. And how far apart the signal and the noise are distributed, in our case here represented by the overlap of these 2 bell curves here. So how far apart the signal and the noise are distributed will tell us about the difficulty to make a good choice. If they are far apart, if the distance are greater, so the choices will be easier. But if you have high overlap between these two, the signal and noise, we have a more difficult situation. Hard choices will be made. So, and it's here where the plastic and pre-resemblance has a role in our theoretical framework to understand plastic ingestion. A key point is to understand how the cues overlap. In our case, cues emitted by plastics, the noise, and cues emitted by food, the signal, right? Where high overlaps occurs means higher resemblance. When we have higher resemblance, it will lead to higher plastic ingestion. But we always need to have in mind that we must think about the sensorial and cognitive world of the studied species. Not our own world, not plastic bags and jellyfish, but what really the animal are perceiving from the environment, right? Therefore, pre-resemblance is very difficult to evaluate in the real world. Sometimes we have some proxies to evaluate that, but it's not an easy thing to do. Back to the theory. Now we will talk about the acceptance threshold. It's the second key ingredient to understand plastic ingestion. And this threshold is about the Q level at which switches from rejecting to accepting an item, right? Low threshold means that an animal will accept a great variety of options, which may increase the chance of the acceptance of undesirable options, right? But at the same time, the animal will be less likely to miss an opportunity when a good option is presented. So, and by sliding the acceptance threshold back and forth, trades off between one error for another, like accepting a low probability option or rejecting a high probability one. So this balance, it's important to understand, right? Now let's apply this to plastic ingestion. And this where a low threshold will increase the risk of plastic ingestion, as you see in the first figure, right? If there is a low threshold, animal will be more prone to ingest plastic. While in a high threshold, the bottom figure will decrease the risk of plastic ingestion. Sliding acceptance and threshold back and forth trades off increasing or decrease of chances of ingesting plastics. And two main factors are related to sliding the acceptance threshold in the plastic ingestion. So let's bring this theory to the real world, right? And pre-selection is one of them. Because generalist species are known for ingesting a broad range of food items, most accepting all encountered prey when foraging, which means they show a lower threshold and consequently will be more prone to ingest plastics. This red line that you're seeing in the figure. In the other hand, specialists are known for consuming a narrow range of food and are likely to accept only the most profitable items, which moves the acceptance threshold to a position that make them less prone to ingest plastic. This scenario is true if you consider the same overlap between plastic cues and food cues. So we need to remember the first piece of this theoretical stuff, that's the overlap. If the overlap is the same, you have this, everything that I said before is true, right? And of course, we can have situations where a specialist will ingest high amounts of plastics due to the high overlap between cues and food, cues from plastic cues and food cues, right? So the overlap of the cues and the acceptance threshold will play a role. So things become to get more complicated right now because we need the interaction of these two parts of the theory. Another aspect, another factor that will influence acceptance threshold is the nutritional states of an animal. This is the nutritional states. This is a factor that will also slide in the acceptance threshold back and forth. And this is interesting because highlights the importance of the circumstance to understand plastic ingestion. Because we may have the same species, the same individual, but the acceptance threshold may vary from the same individual depending on their nutritional state. If the animal are in starvation can induce poor forage performance. increasing the chance of ingesting plastics. On the other hand, healthy animals will be less prone to ingest plastics. So we have one thing that's broader. It's the generalist specialist range, and it can be classified as populations or species, and of course in the individual level too. But to have the nutritional state that is more restricted to the individual level, and it's also being influenced in the chance of ingesting plastic. So here it's a figure that's not very easy to understand, but summarizes the relationship between acceptance threshold and cues overlap. And as this presentation will be available for you after this meeting, I think it's good to come back to this figure and take a look if you want to really understand the theory behind the plastic ingestion. So let's come back to the figure. As we have different scenarios here, range from the lowest risk of plastic ingestion in blue, right? When an animal has a high threshold and there is low cues overlap. For example, this blue species, hypothetical species, can be specialists whose food items have low resemblance with plastics. And the other extreme, we have the highest risk of plastic ingestion in red, when an animal has a low threshold, a generalist, for example, or an animal with a risk of starvation, at the same time that there is a high cues overlap between plastics and food item. Okay, but what's the implication of all this theoretical stuff to the real world? Understanding the drivers of plastic ingestion, the drivers that we propose here, will allow us to develop an evidence-based risk assessment of plastic ingestion. And in this figure, you can see how the drivers of plastic ingestion may interact, not the theoretical one, but the real-world one. And plastic viability is coming to come back to the figure right now. So from the lower risk in light blue to the higher risk in dark blue, and all these black dots are organisms, maybe individuals. So this is the framework that we must have in mind when we're thinking about the risk of plastic ingestion of a given species or a given population, for example. So it will be great to see this theoretical framework that we proposed tested by experiments in laboratory and field experiments, and more than that, this framework can be used to develop risk assessments of plastic ingestion. So one important thing here that we need to have a good knowledge about the ecological traits of the species or the population that we're trying to understand the plastic ingestion. And of course, we need to understand how is the plastic available for this species in the environment. So now for the closing of this presentation, I'd like to highlight that although chronic effects of pollutants are hard to estimate in natural environments, we have some studies showing these effects. For example, one study that I think it's great. The chronic exposure to PCBs has left a worldwide population of killer whales at risk of extinction in the coming decades. So the exposure to PCBs alone, and the models are predicting that some populations of killer whale will become extinct in the coming decades because of the amount of PCBs in these tissues, right? So this study and others provide unsettling examples of how years of conservation efforts can be severely compromised by diffused threats, like pollutants. Right? It's everywhere. It's difficult to understand its impact, but the impact is happening right now. And it's possible that some of the threats to wildlife posed by exposure to plastic pollution, for example, are yet to emerge, right? For example, we have a very good paper that was published last year too, that have some models showing a possible decrease of sea turtles population due to plastic ingestion. It's a model of ecological breakpoint model, and which was using a realistic levels of plastic ingestion. So we're beginning to figure out all the the population level effects of plastic ingestion that can be maybe realized in the next decades. So come back to the one point that I mentioned, the first part of this presentation. We always need to have in mind that we live in a world where the co-occurrence of multiple anthropogenic stressors is the norm, right? And the risks posed by the plastic ingestion trap present here must be examined in this context, right? Not alone, because we have a lot of things happen and we are in the middle of an environmental crisis. So one question that remains is, can the plastic trap be disarmed? It's a question that may help solve this problem, right? And we know that three processes may help to disarm evolutionary traps: natural selection, animal learning, and human management. But I don't have good news for you. Due to the nature of the plastic ingestion trap, we don't have evidence suggesting that natural selection or learning will provide a mechanism for a population to respond timely to plastic pollution. For example, natural selection takes generations, and we cannot predict the outputs in relation to plastic ingestion. And there's a lot of different species ingesting plastics. And the ability to learn and pass on learned behavior varies greatly among species. And then another thing is important when considering learning. It's the temporal disconnection between the act of ingesting plastic and their negative outcome. Because most of the outcomes that we know, the negative outcomes of ingesting plastic are chronic in its nature, right? Animal will eat, will feel nothing at the time, but when plastic are accumulating in the organism, now you have the effects. So this disconnection between the act of ingesting and their negative outcome decreases the possibility for an individual to obtain a feedback on the poor payoff of plastic ingestion. So learning is very difficult to help us also in this question. What leads us to human management? In this case, reducing the encounter rate with the trap, which means reducing the amount of plastics in the environment. It's not easy to, but it's the only option that it's really feasible. So, which maybe, and how can we do that? It may be achieved by addressing the whole life cycle of plastics, right? We need to think, every time that we think about plastic pollution, we need to think about the whole life cycle, not just the end of the life cycle, where the plastic is already in the environment, right? So we need to decreasing plastic production and use, shifting toward the circular economy, and increase the investments in waste management and recovery. And these aspects are the key aspects for the core goals of the plastic agreement that is discussing right now in the UN, right? So thank you for your time, and here's my contact if somebody wants to write me about these ideas. Thank you again.
Thank you, Dr. Hobson, for the important shared information. Now, the chair, Professor Raymond Ward, will be conducting the participants' questions to the speakers.
Yeah, hi, thanks for that, Louisa. Hopefully my microphone is working fine. We do have several questions, and then if no others appear, I've probably got some to add in as well. First off, thank you very much to the three speakers, three excellent presentations. I think we'll probably start in order because the questions seem to come in order. I think this one was for Sedat. It said, do you not think that it might be a greater priority to focus on alternative methods of developing plastics in the short to medium term, e.g. kind of biodegradable materials?
You asked me, Raymond?
I didn't ask, it was Francisco, but yes, I think that was the question.
Okay, no, I mean, you directed the question to me, yeah. Okay, so the alternative materials very long time, we know that this kind of materials can be considered as an solution for plastic pollution. But in reality, unfortunately, such kind of alternatives are not really capable to be a really alternative for petrol-based plastics, because the limitation of the degradation and their performance in the natural environment is not really different than conventional plastic. So we also know that all this bacteria, fungus, and other kind of potential degraders are not really capable to degrade of all this kind of plastics, because we have hundreds of different kind of plastics in the daily life, and thousands of chemicals added inside. So I think we should focus first on the reduction of plastic production rather than finding an alternative. So if we continue to produce such amount of plastics, so any alternative can solve anything about plastic issue.
I will probably add to that because I put it in my questions. I think that's a good point, that probably reduction is one way to deal with it. But do you not think this needs to be a multifaceted approach to dealing with the problem, i.e. biodegradable materials are probably part of the solution? Probably Colin can jump in as well, because he kind of works more with the kind of degradation as possibly part of the solution.
Yeah, I'm happy to chime in. I agree 100% that there needs to be a multifaceted approach. I mean, there's no doubt in my mind that reducing consumption is a very logical and should be an easy path forward. The challenge with that is humans are extremely resistant to change, right? So demand is not going down anytime soon, regardless of regardless of the fact that it should go down, right? Demand is going to continue to rise. And with that demand comes or necessitates formulations that are actually degraded in the environment. And there's lots of examples of bioplastics that degrade in the environment. So, there's lots of great studies on PHA degrading in the ocean. We've written several papers now on cellulose diacetate formulations degrading in the ocean. And I would actually hope that the community would focus a little bit beyond just biodegradables. Thinking about alternative means to shorten the lifetime, whether it be adding in, photocatalysts that can break down plastics faster in the environment. Coming up with ways to develop this next generation of materials that provide us a solution on the short term, while these sort of fully recyclable, chemically recyclable, circular economy materials are being generated.
Thanks a lot. That kind of leads us a little bit on to the second question, which is from Jones, and that was, would the problems of plastic pollution disappear if we stop producing or using plastic starting today? Can we say that there is a viable solution to end this environmental problem? And from an economic point of view, would using bacteria that break down plastic quickly be a viable solution? Which kind of leads back onto what we said before. And I'll probably leave that open to all three of you, and I may chime in if needed as well.
Okay, if you think about the ingestion by the organism, no. If we stop to produce right now at this moment, there's no plastic anymore. We have a lot of plastic in the oceans. So the oceans, the land, so the rivers. So the problem is not stopped. It will not stop. I think it will be a problem like the climate change right now. We need to manage this problem, to have a goal to reduce, but we will always need to deal with the problem. I don't see this problem go away with just minimizing the impact of the problem, I think.
I mean, that kind of goes back to what Sadat was saying, that we've got these huge kind of stores of plastic all over the place, and many of them have been shipped to countries that have kind of less strict legislation to limit the kind of transport of those plastics to the environment. So yeah, I suspect you're right. We're probably even if we just stop today, we've got this problem ongoing, and I agree. I think it's probably that managing part of the problem rather than than that. I did have a question that kind of leads on to something from Siddharth. I said there's obviously an economic driver for export of plastics to other countries, and my question to you then, and well, to any of you actually, is who do you think is responsible for controlling that? Is it the countries that are exporting? Is it the countries that are importing? Or do you think this maybe could be dealt with on the international stage?
Yeah, both sides. Yeah, both sides, but the most the exporter side, because if something is recyclable, then you can recycle in your own on territory. So it's not necessary to send it countries which can even cannot manage its own plastics. Just think about this importer side. They don't even manage their own plastics. Almost 90% of the municipal waste of this importer side are going to be landfill. So they even cannot, recover all those plastic waste that they generate. But still, the exporter side pushing these plastics to those countries because there are brokers and some operators, illegal operators, facility owners. So they are making agreement each other, and then this waste flow happening. So both sides have responsibility on this, but mostly exporter.
Okay. Anyone else have any other views or want to jump in on that one?
I think I just have a point that we need to rethink the concept of recyclability. Because if you have the plastic in your country and you are not able to recycle there, or because there is no the facility or the technology, that plastic is not really recyclable. So we have this label recyclable, but we know that just a little bit of all the plastic products are recycling. So we have a lot of years that the industry has put this concept of recycling for the society, but in practice, most of the plastics are not really recyclable in its whether you're consuming or we don't have enough facilities to recycle all the plastic produced. So I think you need to rethink this concept of recyclable because it's a key component of how we will deal with this problem.
Thanks a lot. That was good, and it's nice to get some kind of broader response on that question. I've got some additional questions. There's one from Graziele to Colin. It says, and I'm going to add to this as well. It says, in general, in tropical regions, do you think decomposition of plastic is more or is quicker? And my question also to kind of add to that was, has anyone done any studies on different light spectrum and how that actually degrades plastic? Just because obviously you got different light spectrum at different depths. So I was just wondering if you knew anything along those lines.
Yeah, when I think about, the lifetime of plastics or the decomposition of plastics and how they vary spatially, I think you have to consider, of course, the environmental variables, but the actual formulation of plastic matters a lot, right? So we need to really think about, well, what's the polymer backbone? What's the molecular weight of the material? What are the additives that are in it, right? What's the surface area of the material and how that evolves over time. So it's tough to just say, okay, well, they're going to degrade faster in the tropics. One thing you could say is that, sure, from a biodegradation standpoint, it's warmer in the tropics. So you'd expect that biodegradation rates would be faster. To get to your point about sunlight, there are some studies looking at the wavelength dependence, right? Generally speaking, pure polymers like polyethylene, polypropylene, they don't degrade much, or they don't absorb much in the UV. They only absorb the UVB light, right? So if that material, gets, let's say there's a wind current that has vertical transport in it, and it pushes the material to a meter below the sea surface, a lot of those UVB bands are going to be attenuated already. On the flip side, most consumer plastics contain all sorts of additives that absorb broad amounts of light, right? So we're finding when we do, when we analyze the absorption properties of consumer plastics, there's quite a bit of absorbance throughout all of the UV bands. And of course, a lot of plastics are colored, right? So they absorb in the visible region as well. So our lab is pretty focused right now on thinking about, how the degradation rates would vary as a function of water depth, with the understanding that blue light, penetrates the deepest into the water column. So if you can design plastics that absorb blue light efficiently and degrade efficiently in the blue, that'll definitely shorten the lifetime.
Brilliant. Thank you very much. I like that answer as well, because I got something new there as well. Samuel, and I guess this probably maybe is one of our last questions. He's put a very big question to the panel. With the issue of climate change and plastic pollution putting more weight or burden on the marine environment, coupled with population growth, is there a future for the marine habitat? And what conservation mechanism can be put into place to reduce these threats? It's a big question, that one. Not sure whether any of us could possibly.
Could I just generally make a comment about it, maybe?
Yeah.
One thing I think we need to do better as scientists is think more broadly about which threats are most important for ecosystem and human health, right? There's a lot of, I think there's a lot of misconceptions about which threats are most important or which threats drive. And it's not only how the threats work independently, but also the synergy between the threats. So I'd like there to be more discussion about sort of prioritizing the different threats to these ecosystems, and then thinking about the impacts, right? Because right now there's, how do I say this? Sometimes I feel like the media drives the perception of the threat, whereas the reality is the scientists, we don't really know to the extent that all these how serious a lot of these threats are, and then how much they work together and have a synergistic threat to ecosystems. So it's hard to answer the question until you know which threats are most important.
Hobson or Siddharth, did you want to jump in there as well?
Yeah, of course. And of course, we have a future for the marine environment, but we need to choose it to be a good future or a bad future, but the future will be. But now it's really a tough moment, but you need to realize that we have a lot of that we can do about this. So I don't think that we need to have this doom view of the world. Of course, things are getting worse and worse, but we need to act, and everything that we act will change the course of the future that we want. And jumping in the calling comments, I really think that we as scientists need to have a more broad view connecting these global problems, because we have, there is climate change and there is plastic pollution and there is biodiversity crisis, but all these crises are interconnected. And sometimes by solving one, you can solve another. So I think this is an important thing, that we as a scientists are not discussing the connection between those. But sometimes you can see like, climate change is more important than plastic pollution. But acting on plastic pollution and will help to act on plastic and on climate change and vice versa. We just need to have a more broad framework to think about these global problems. Because today my view is that a lot of different boxes, and it's not the real world. The real world is always connected, So I think we need, and by connecting these dots, we can also have more efficient conservation acts, for example, because plastic pollution will call the attention of the public, for example, more than climate change sometimes. But we can go through there, connect to the community, and come back to the actions that will acting on both climate change and plastic pollution. So I think we need to get out of our boxes in science and try to connect things.
So I guess that's kind of the main take home from that is that we should probably not sit in silos and try to work across broad, kind of dealing with the broader environmental kind of crisis. We've got one final question, and then I guess we're going to have to wrap up because
may I add something?
Yeah, sorry, Sadat, go on.
No worries. Yeah, I totally agree with Robson but I should add something. There is a cause and effect relationship between climate change and plastic production, because when you produce more plastics, then you are contributing climate change, and then it changes meteorological events like heavy rains, like abnormality on the rain and other things, and it causes flash floods and this huge amount of rain just dropping in one time in any part of the world, and then all those plastics that we couldn't manage in the land, in the terrestrial area, flowing into the Mediterranean Sea, like in oceans, in other part of the world. So there is a cause and effect. You are promoting climate change, and then climate change promoting marine pollution because of flowing all this plastic waste from land to sea.
Excellent. Thank you, Sadat. There is another question, but I think because we've overrun quite a lot, I'll try and wrap this one up. If possible, could the speakers just maybe answer in the question and answer section there, or on that part there, and I will probably pass you back over to Louisa to say what's going to be happening next. Okay.
Thank you all. It was a very interesting discussion, which added a lot of knowledge to everyone in here. The questions that weren't answered here will be answered via email. Now we'll have a quick break of 15 minutes, and 1:30 on UTC time and 10:30 at Brazil's time, the session 2 will begin. So see you in a minute. We are back, and session 2 has as theme how to do research in plastic pollution. The chair of this session is Dr. Raymond Ward, visiting professor at the Colégio de Estudos Avançados at Federal University of Ceará, and professor at the University of Brighton. And the speakers are Dr. João Frias, Dr. Filipa Bessa, and Dr. Humberto Andriolo. In the second session, there will be all 3 speakers' presentation, and then a 20-minute discussion where the questions from the audience will be answered by the speakers. First, I would like to introduce Dr. João Frias with a presentation entitled Monitoring Microplastics, Sampling Methodologies for Surface Water and Benthic Sediments. He completed a bachelor's degree in environmental engineering, a master's degree in ecological engineering, and a PhD in environmental studies, focused on marine litter, microplastic, and ecotoxicology. He is currently working on the JPI Ocean Microplastic Project and is based on the Marine and Freshwater Research Center in Galway on the west of Ireland. So welcome, Dr. João Frias, the screen is yours.
Hi, Louisa. Thank you so much. I am going to share my screen here, and please can you let me know if you are able to see the full screen presentation? Yes, all good?
Yes, we are seeing.
Okay, great. Thank you so much for this opportunity. First and foremost, I would like to thank the organizing committee of this event for inviting me to participate. my introduction was already pretty clear, so I will jump into the talk, where today we're going to see how we can survey and sample different environmental compartments. So in my session, we will go through a little bit of background information and some concepts, because on the morning we were already like speaking a little bit of some of the terminologies. However, I wanted to make a couple of points clear. And then we'll dive into the methodologies for surface water and benthic sediments, because my colleague Felipa, she will be addressing other sampling environmental compartments. I will also speak a little bit about the interface and what that means, intertidal coastal zones or something else, and some take-home messages. So in terms of background, I am sure that maybe images like this are might be a reality in your local beach, depending where you reside in the country. For me, this image speaks volumes because it is the beach that I used to go with my family when I was young, and this was the reality when I was in college, when I was in university. So this photo is kind of the reason why I am in this field, and why I decided at the time when I was still a master's student, under supervision of Professor Paulo Sobral in Portugal, I started working like in this topic. And at the time, like really early in my career, I do remember that Professor Monica Costa from Pernambuco came to my university to do a small lecture on plastic pollution and microplastics, and I was really interested. I thought it was really inspired, and maybe like these two women point me out in the right direction on where I am right now. And I hope that this training course today is also inspiring for you in your personal and professional careers. So, how did we get here? And thanks, Sadat, for pointing out a little bit on what we've been doing so far and why we are in this problem. Why do we have plastic as an environmental threat and as an environmental issue? I think it all started around the 1950s, where plastic production at the global level started. You can see this article from Time magazine approximately 5 years after plastic production at the global level had started. And we also need to understand the historical context of an image like this, because this image says that disposable items can cut down household chores, meaning the time that you would spend doing your household chores. And this is a couple of years after World War II. There was a need of you spending more time with your family and with your friends because of this scenario that had happened. And there might be one of the reasons why planned obsolescence was kind of developed around more or less of this period. What I wanted to point out is that plastic production started with 1.7 million tons being produced in around the 1950s, and in 2021 it reached 367 million tons. As you can see, it only decreased doesn't decrease really regularly. It actually increases at a rate of 8% a year exponentially. To my recollection, it only decreased in 1973 during the oil crisis in the Middle East, between 2008 and 2010 with the global financial crisis, and it is decreasing now, or at least it's keeping stable with the COVID-19 global pandemic. The red plot, you can see the cumulative plastic production, and you can see that it's in billions of tons, not in millions. So it's 9.5 billion tons, considering that human population also reached 8 billion individuals in the planet this week. It is something for us to consider how we consume. There is an interesting timeline, and I think that in a sense this goes with what was being discussed in the previous panel. So 1950, plastic production started. We started having the first evidence in the environment. The first paper that I remember was 1969. The most famous one is 1972. 1973, we had the oil crisis, but you see there are a couple of important relevant protocols, either at the UN level or other relevant institutions that kind of focus on agreements. I wanted to highlight the Kyoto Protocol on climate change and how much has passed since then. But also strategies that have been developed for plastic pollution, like the Honolulu strategy in 2011. I also want to point out that 2004 was the first publication on microplastics. And how does this relate? Because this was one of the things that was being kind of in discussion from the last panel, is how this relates to CO2 emissions and to the volumes of plastic being produced, but also on the publications on microplastics. And you will see that there is quite an exponential increase over there, because this concept was used on the context of plastic pollution for the first time in 2004, but we will get to there. Also, I want to point out that our systems are evolving and are changing. We used to find a lot of natural items in the beaches, and now we are starting to see a lot of different items that are left there by mistake. But how this is changing the global game. In order to be a little bit clear and a little bit to the point about what I'm going to talk on these techniques, I wanted to share with you the concept of marine litter as it is envisioned in Europe under the Marine Strategy Framework Directive, which is any persistent, manufactured, or processed solid material that is deliberately or unintentionally discarded, disposed of, abandoned, or transported by winds, rivers, and animals to the marine and coastal environment. And I wanted to thank Colin for pointing out the extraordinary work from Chelsea Rockman. On 2019, my team also proposed a definition of microplastics, and at the same time, Nana Artman in Denmark proposed a very comprehensive set of criteria to evaluate and identify what a microplastic is. Here in our lab, the definition that we use is any synthetic, solid, particle, or polymeric matrix that is regular or irregular shape, that ranges in size from 1 micron to 5 millimeters, of either primary or secondary origin, that is insoluble on water. And we use this kind of definitions in order to narrow what we are searching for. In terms of origin, there is a distinction between primary and secondary microplastics. The primary ones are the ones that are designed and manufactured to have microscopic dimensions, like the ones that are added to makeup, to exfoliants, to toothpaste, and other personal care products. And the secondary microplastics that result from the fragmentation of larger plastic items while exposed to weathering conditions in the environment. So for this specific work, everything started with a publication from Valeria Hidalgo Ruth from Chile. And basically, in this study, they compared 68 peer-reviewed studies that were assessing sampling methodologies, number of samples collected, size meshes used while sampling, and other categories to identify microplastic. It was published in 2012, if I remember correctly, and it was quite an extraordinary new piece of evidence when we were talking about the lack of standardization methods, and also on the lack of standardization methods. In between 2016 and 2019, there was a project in Europe from JPI Oceans, funded by JPI Oceans, that define baselines and standards for microplastic analysis. And this is how or where my journey into the harmonization of sampling, lab processing, and analyzing of microplastics starts. So it started kind of with the difficulties in comparing data. We still don't have standardized way of reporting units. So it was kind of like comparing apples and oranges. Sometimes it is very difficult to process this kind of samples because it's like finding a needle in a haystack. And then sometimes our focus approach is still very under a university point of view, or like an ivory tower, when it needs to relate more to practical monitoring. And we need to see also how the monitoring can fit into policies. So diving into the methodologies. There are a couple of different environmental matrices that within my team we sample: surface water, benthic sediment, biota. We're kind of like going into fresh water, assessing also how climate change or extreme weather events actually influence accumulation. But today I'm only going to focus on two. I'm going to focus on seawater surface and on sediment. So on seawater surface, usually there are a diversity of techniques that can be used. Probably the most common method that you've heard is the manta trawl. This is a method that mimics the feeding strategy of manta rays. And basically what we do is we have a large net with the specific size mesh and a cod end where everything is going to be accumulated and where we are going to collect our sample. Of course, because we are sampling at the surface, we will collect a lot of zooplankton, and that might represent a challenge to what we want to do further with our analysis. So as I was saying, there is a diversity of techniques. So you have the manta trawl, you have the catamaran, you have the Newton net, you have the Avani net or the Avani trawl. These are all kind of options. If you want to focus on one specific aspect, let's say that you are doing zooplankton and plastics will be a secondary thing, you can use a method like this. However, if you want to have like clear distinction between the two methods, my suggestion would be to use, for instance, a bongo net, where you could use the same size mesh, different cod ends, and in one you would use, for instance, for zooplankton, and the other one you would use for plastic pollution, so that you can follow different methodologies. Because usually for zooplankton you need to preserve, sometimes in formaldehyde, and for plastic, because it will be the items will be there, you can just freeze your sample. And there's this would be much better if you wanted to analyze both, because it will be more costly in terms of time to deploy a manta twice. You have other methods to sample at the surface. In a vessel, you can use Van Dorn samplers. You can also do it in rivers. This is a Schindler-Patalas plankton trap, which can also be used in freshwater systems and in seawater systems. And then while you are at sea, you can also use the subsurface if you want to go not really at the surface layer, but a couple of meters in depth. And for that, you can use different pumps, either displacement pumps or dynamic pressure pumps. Or you can even try, if the captain of the vessel would allow you, to tap into the systems that already go through the vessel and try to filter water directly from there. What we do here in Galway is, for instance, this is a relatively recent example where we went and we deployed 20 manta nets for approximately 20 minutes at a speed of 2 knots. And basically we were doing these maps of microplastic abundance and how these relate. We always put the results in a way that the majority of people can understand that we had mainly fibers and that the two main colors were blue and black, and you can see the densities. And of course, if you go to the paper, it will have a more in-depth explanation of what we did. One of the highlights of this paper is that this was the first time that we actually recorded primary microplastics, so microbeads, the ones that I was telling that might be in your makeup or in your toothpaste or in any other personal care product that are intentionally added to personal care products. We found 2 of them in our samples. Now, some of considerations that we need to take into account for surface waters is the different size meshes to sampling. And for this, I will go back to the paper from Valeria. And another aspect is something that Giuseppe Muswaria pointed out in a paper from 2016 on how we do to actually distinguish by using a GPS or using a flow meter or using both in order to have a better assessment of our sampling. Another consideration, but this is more into processing, is if you have samples that you want to have like correct aliquots, you can use the Motodo or the Folsom plankton samplers so that you can have separate samples that will have more or less the same concentrations of everything. And then you can use one aliquot for zooplankton and another aliquot for microplastics. When it comes to benthic sediment or sediment in general, these are some of the most common techniques that you can use. You have Van Veen grabs, box scorers, petit ponard, or a day grab. You also have other techniques such as a multi-corer, and depending on where you are of what kind of access you have to technology, you can use remotely operated vehicles where you can go and use these robots to collect benthic samples for you at high depths. Ideally, what we do in our work, because it's slightly shallower, is we use box scorers. And this is one of the techniques that it's quite good. So you would see that the box scorer would be deployed to the sediment. It would create this kind of box, and when it closes, it maintains the structural integrity of the sediment in there. And what we do here in Galway is we actually use a separate metal corer, and we subsample within the sample that we take the first 5 centimeters so that we can have a little bit of variability inside of the sample, but we collect enough samples to do similar exercises to the ones that I was showing before, where we can see where the highest concentrations are. And again, we try to relate this into ways that people can easily understand by making these heat maps. This is a work that my colleague, Dr. Elena Pachter, did quite recently. And she identified that in the benthic sediment of Galway Bay, the majority of microplastics that were being retrieved were fibers. Now, there is this distinction, like I was saying, or this like connection between between the interface areas. And for here, you can actually look at it from an intertidal perspective, or you can use this kind of techniques to sample other things. Like, for instance, you can use an ice corer and then melt the ice to search for microplastics, which has been used even with people here in our team in Ireland. And of course, you can look at it from a different perspective, but I'm not going to go in depth into here because most of these things are already in protocols that are published. to harmonize methods, or already like to give you a way of how to sample if you are looking to benthic and intertidal sediments to seawater surface, or just at intertidal sediments with sheets inside. One of the things that I wanted to point out, because it came also from the discussion before, is these need to prevent or reduce microplastic cross-contamination. So usually what we do here in our lab is we do our filtrations under laminar flow. We always acid wash all our glassware, and after that we triple rinse it in ultra pure water. When we leave it to dry, we'd always leave it to dry with the mouth upside down. On the operator itself, we use 100% cotton lab coats. We always record the color that we are wearing underneath, so that if we are observing something and I'm wearing green today and we suddenly have a lot of green microplastics, we can relate back and see, oh, okay, there might be a bias moment there because the operator was using a color that was the most predominant color on that day or on those couple of days that we were doing the process. To minimize everything as well, we filter all our chemicals and all our solutions, and we always have procedure blanks and controls for the experiments, and atmospheric blanks or atmospheric controls during sampling at sea, during sampling in the lab. Kind of to minimize again and to have like this forensic science approach to it. So kind of to wrap it up and to share with you some take-home messages. Lack of standardization is still an issue. So one of the things that I use when I talk in platforms like this is if you're not sure what unit to report all of them. Meaning, it is important that we have an idea of how many microplastics are per area, per volume, per mass. This particularly for sediment and for surface waters. If you have the chance, if you have the possibility, because you have GCMS, or you can do this kind of like mass quantification of microplastics, then report that in mass per area, mass per volume, or mass per mass of sediment, for instance. The grams or nanograms or milligrams, sorry, of microplastics per kilo of dry weight of sediment in a given geographical area. For biota, I'm not going to go there now because I know Felipe is going to touch on that. So depending on the species, either you go number of microplastics per individual, or you go in number of microplastics per gram of edible tissue if it has a commercial value into it. We need to have holistic approach to use monitoring on our way, on how we use and how we developed our sampling or our collection, extraction, purification, and identification techniques. We need to collaborate more with modelers and also with people developing ecotoxicology. And given, like, at least in Europe, the importance of the new European Green Deal, we need to start also assessing the circular economy, or this import and export of waste, and how it goes around the world. As we've seen today, it can be also a challenge, as we've seen in Sedat's presentation. To conclude, I always like to have like this kind of visions of future, and I wanted to start with how people in the 1960s saw the future from their own perspective. This was like what in the 1960s was envisioned to be a modern leisure area where you could listen to music in your house. But I like this one quite well because this is quite realistic. This is how San Francisco was envisioned in the 1960s. And you can see like the bridges, the skyscrapers, everything. This one I quite like. We haven't reached there yet. I wonder if we are going to reach there because we are going through so many things right now in the planet. And we are in the middle of a climate crisis. And we do need to start acting. Everybody knows the reduce, reuse, recycle. but maybe it is important for us to start rethinking, repairing, refusing plastics in our life, finding alternative ways of actually not getting into this scenario and maybe go more into a scenario like this for the future. We can do that through our research. Our research can contribute to policy making. Baseline data are absolutely important, but our solution need to fit particularly important policy goals and regulations. And on this, you can only achieve this by directly going and collaborating or being part of policy processes like our team does here. And I'm glad to say that in 2019, Ireland passed the Microbeads Prohibition Act, which is starting to legislate on the intentionally added microplastics on personal care products and other uses, but also how you do outreach in schools, in universities, or directly broader way as we are doing here today. And where you go and how you can contribute to global dialogues, how you can be part of the Sustainable Development Goals, not just being something that is highlighted in the United Nations, but how we bring that into our daily lives through how we live our life and what kind of ways we go when even the conferences that we attend, because it is important also for scientists to make this bridge directly with policymakers. Thank you very much for your time. I also want to thank my amazing, glorious team. Some of them have already left, but no longer, even so, they are still part of us. And I wanted to say that this is a massive problem. And just because you don't see it doesn't mean that it's not there. It is important for us to act into this kind of things. Thank you so much for your time.
Thank you very much, Dr. João Frias. I hope you all enjoyed this amazing presentation. Next speaker is Dr. Filipa Bessa. She is a biologist and researcher at MAR, Marine and Environment Research Center, from the University of Coimbra in Portugal. With a background on marine ecology and a PhD in bioscience from the University of Coimbra in collaboration with the University of Florence in Italy, her current research focus is related to the assessment of the effects of microplastic pollution on marine and coastal ecosystems, with particular focus on biota. To this regard, she is involved in several national and European research projects, and she is a member of the International Working Group Micro and Nanoplastics in support of the European Commission Scientific Advice Mechanism. The title of the presentation is Monitoring Microplastics in Biota, Challenge and Recommendations. So welcome, Dr. Filipa Bessa. The screen is yours.
Thank you, Luisa, for the presentation. And I'm going to share my screen at the moment. And well, I hope you are seeing my screen now, right? Yeah, thank you. I would like to start by recognizing the importance of these meetings and to say thank you for the organizing team to invite me to present and to be here today. It's an honor and a pleasure to be here with you today. And of course, that after the Dr. João Frias presentation, I'm still dealing with that. Thank you, João, for your incredible presentation and for the introduction on this topic. I'm going directly to tell you more about the monitoring microplastics in biota and how these was evolving during the last years. So as we all know microplastics, and we have this notion that microplastics are here, there, and everywhere. And every day, news and also the scientific community has been highly concerned about the presence and the widespread of these particles around the globe. But still, microplastics have been detected in a wide variety of habitats, in the ocean, from the shallow coast to the deep sea, and even in remote areas where people are not, able to be there. And microplastics have been detected, in all marine ecosystems, in all terrestrial ecosystems, even in the air that we breathe. So most of these notions have been documented in several platforms. I can show you I can share with you 2 different platforms that highlight the amount of publications that reveal the presence of microplastics in biota. The database is a database that started in the 60s, and as you can see from the green dots, these are the amounts or the level of publications that the scientific community have been produced in the last years regarding ingestion of microplastics by biota. But recently, a great team from Brazil also started a new platform, the GLOVE platform, the Global Plastic Ingestion Initiative. And just for curiosity, I looked into the Brazil level, the studies that have been produced in Brazil, and you can see this platform allow us to choose the level of not only the geographical areas, but also the level of taxonomic species that we want to see and to address. It's a very interactive and very intuitive platforms, and can show us, not only that microplastics are widespread, but which taxonomic levels we know, or the levels that we need to explore in the future. So microplastics, has my previous colleague referred, and also the previous panelists, microplastic pollution is in the public domain as well. And we know that they are different, they are complex, they are diverse. We can see from the extraction of microplastics from the environment that they can present a variety of sizes, of shapes, of colors, of polymers, of additives, or even bacteria and other biofilms that can form in these surfaces. So this diversity of particles can also be very tricky for their identification. And to explore more the relations between the particles and the effects, we need to monitor, we need to know how the world is going with this diverse and this sometimes beautiful particles. But why we are concerned about them, why microplastics can be a problem? It's not only a question of the plastic per se, as we know that they are not, the polymer per se are not a problem, since physical blockages can occur, but are not that, they are not the majority of the problems related with microplastics, but the additives that they contain, and they are really, most of them soluble, and they can be present in the water, and they can also be absorbed by most of the species that we know that can receive these amounts of additives. But not only the additives, the pathogens, the biofilm that forms microplastics after 2 or 3 days in the water can be or can contribute to some diseases that we know that are in those species. So microplastics are not only a problem regarding the polymer, but the entire level of typologies that you can see here: the polymers, the additives, the prototypes, the sizes, the morphologies, and all these materials that can be attached to their surface. The interactions with biota occurs also in the microscale. We know that in lab conditions, microplastics can be ingested, and of course can be a potential for some diseases. However, these effects are mostly recorded in laboratory conditions. We still have some doubts about what is happening in the natural environment and the implications that those particles can have in the entire food web. And these international conditions are still not well understood. We know that they are still there, they are persistent, but we are, well, the story is still incomplete regarding their effects in the natural environment. So the main questions related with microplastic pollution in biota are still evolving. We are still looking at the levels of microplastics in the species, in the natural systems. We are still looking where are the hotspots of microplastics in the natural environment. We still just know the effects to species in laboratory conditions, in scenarios most of the time completely different from the natural ones. We still need to know the long-term effects, how these can be associated with the effects to human health, and what are the solutions for microplastics and what we can do in the near future. So these are mainly one of the most important baseline for the importance of monitoring microplastics in biota. We know that with this information, we are providing environmental data essential not only to know the sources and pathways of those particles, where are the hotspots, as I said, what are the levels of toxicity, and if there are risks to humans, and the entire risk assessments of those particles, which are clearly underrepresented in the scientific literature at the moment. And within this in mind, we, as João referred, we participated in an international project, BASEMAN project, with the goal to prepare a set of protocols for harmonization, the monitoring of microplastics in different compartments. And me and a big team prepared the protocol for monitoring microplastics in biota. And I'm going to share with you some tips if you are interested in working in that area, because as we know, and as other panelists referred, there are still very inconsistencies in the data reporting, in the methodology, in the methods, and we need to start focusing on this, and by sharing the best practice and sharing some recommendations. And my talk will be devoted to these particular steps. How we start. If you are interested in working and in monitoring of biota, what are the steps for conducting a real monitoring plan? Of course, that we need to have in mind that these are important statements also for the goals that we have for at the global level. So we know that we need to reduce marine pollution. So this information can fulfill a lot of recommendations that we have in our, let's say, international and national agendas. So this information feeds also these targets and these important agendas that we have. The first step is, of course, selection of species. If you have in mind to work with microplastics and select species from the aquatic environment or from the marine or freshwater, which species could be important to monitor? And this can be, quite simple if you just select a species because is important for fisheries or for human consumption, but we still need to take into account other characteristics of the species. A natural species or a bioindicator species or a sentinel species of plastic pollution should be a species with high abundance and a wide geographic distribution. It should be a species that are easy to sample and process in the lab. For that, macroinvertebrates are commonly selected for these particular studies because they are easy to sample and they are widespread. So species that are already used as bioindicators for other contaminants in the aquatic ecosystems can be used as well for plastic pollution. And of course, the importance of ecological and social ecological relevance, it's also important if you want to establish a correlation with human health, and species that cover several functional roles or feeding yields. For our experience, we suggest a sample size of about 15 individuals per research unit. Has most of the studies sometimes use a lower number of individuals, and it could be not feasible for extracting big conclusions about the microplastic ingestion. It's important also to ensure that monitoring of these species we take, if you select, for instance, the fish, that you extract the full gastrointestinal tract, or at least you select parts of the entire gastrointestinal tract. But for instance, if you use bivalves or small species, the entire animal should be examined. Of course, that if you have in mind studying other species and we want to perform non-invasive methods, it could be also feasible. We sometimes use the scats of the animals if you are working with endangered species or iconic species, or even seabirds, and most of the time these can act as a proxy of ingestion of microplastics and are also valuable for monitoring those particular species. I'm referring, for instance, several top predators are mainly used for monitoring by using non-invasive methods. The sampling methods, it's also important to just take a look around or use a set of characteristics from the sampling site, because most of the time we can correlate the levels of microplastics that we have in our species with the landscape, with the environment. So the time, but also the importance of collecting potential sources of microplastics are indeed important to have the entire snapshot of the environment that you are able to work with. And in general, the approach in the lab follow three main steps. We select the species, and according to the type of species, several steps should be performed. So you need to extract the gastrointestinal tract or use the entire animal if you choose a small macroinvertebrates. If you can run in parallel water and sediments samples, it could be really interesting, because for selecting a bioindicator species, it could be useful to have also the levels of the environment around the species to correlate and to associate these levels. The main step, it's common, a chemical digestion. There are a lot of protocols that you will found in the literature, but the most common use basic solutions, and then we filtrate this solution, and then the last step is extracting and analyzing the particles that you can extract by these different steps and these different analysis. This the processing and what should be the solution that we should select. This is very it's not there. one solution for all type of biota. So in our particular case, we tested different acid and basic solutions, and we have at least some recommendations according to the type of samples that you are working with. If you have samples with a higher organic content, then you need a more strong extraction method. So in our protocols, you'll find a lot of suggestion according to the type of samples that you are working with, what are the best solution, the fast approach, and of course, what are also the protocols or to avoid, for instance, the acid digestion. We know that it can affect the polymers and are not recommended for this type of studies. The contamination control is also I would say, the most important part of studies regarding extraction of microplastics from the environment. Has João also mentioned, not only all the materials and equipment should be washed and the entire process should be completely controlled. So we should assure that negative and positive controls are placed in during all the processing, not only from the extraction, but also from the last part of the analysis. And at the end, we came with some filters. They look like this, more or less, according to the type of your sample. And we identify all small microplastics or particles according to the color, the shape, the size, and the polymer type. And then we quantify the levels of microplastics per individual. It's the most common unit, but we can also use the mass of the individual or of the organs, depending on the fraction that you are working with. And the final step is the identification of the polymers extracted. The most common methods are in the field of microplastics are the FTIR or Raman, spectroscopy. Pyrolysis is a destructive method, but indeed very feasible for the analysis of microplastics. And at the end, we came up with a spectrum that we compare with a baseline, and we can confirm the synthetic polymers that are in the sample. And this is also important because the first studies analyzing microplastics in biota, they just identified the particles by visual inspection, and now we know that well, sometimes most of the particles are also from natural origin. So it is indeed crucial to identify and to proceed with this step at the end of the protocol. And we should report in the standardized way, at least we should report the particle counts with confidence intervals, the detection limits of our particles, with what are the minimum particle size, and at the end, the polymer types and the percentages of particle sizes should be reported for the individuals that we are working with. This is just the first step for a complete risk assessment in applied microplastic pollution research. So we still need to struggle and detect microplastics in several species, but we still need to know what are the plastic tracers. So what are the additives? What are this footprint in the animals? Then we should look at the biomarkers detections as well to infer the health of the animals, and if there is a connection between the plastic ingestion and the plastics tracers, and how they evolve in their health. Then with this information, we can start working with thresholds, and at the end, starting mapping a risk assessment for the species and ultimately for the human health that is always point out as an urgent matter. So as a final remarks, if you are planning to work with microplastics and in biota, the first thing is to define correctly your scientific question. Are you monitoring microplastics in biota? Should I rethink the species that I need to work with? Is there any other studies in the area that I will conduct my study? Explore and harmonize the methods, use the previous ideas that we shared today with you, and of course share the data. It is really important to share this information, not only to have the scientific information, but to help the policymakers in their activities to mitigate microplastics. And this has been done currently in some parallel activities. I share with you the SAPIA that is working directly with the scientists, and the information we cannot only apply and only ask for better solutions and better policies if we do have data and baselines, and this is crucial for mitigate as well and for communication issues regarding microplastics in nature and in the society. And that's all from me at the moment. And if you want to chat with me, or I'll be happy to share with you some thoughts. Yeah, these are my email, and thank you for your attention.
Thanks to Dr. Filipa Bessa. It was a very important theme to be discussed. The third speaker is Dr. Humberto Andriolo, and he will be presenting Beach Litter Survey by Drones, Operational Framework and Potential Outcomes. Humberto Andriolo has a PhD in External Geodynamics and is based in INEX Coimbra, Department of Electrical and Computer Engineering, University of Coimbra in Portugal. His expertise includes development and applications of numerous remote sensing techniques for environmental monitoring. Together, Gil Gonçalves and Humberto Andriolo have developed the UAS for Litter project, which aim at drawing up a drone-based framework, software and hardware, for mapping marine litter on beaches and dunes. So welcome, Dr. Humberto Andriolo. The screen is yours.
Yeah, good morning, everybody. I hope you can see already my presentation. So today I'm talking about the beach litter survey by drones, operational framework and potential outcomes. So as said, these works have been developed within the WASP for Litter project. And today, Professor Gil Gonçalves is not available, so I will present, I prepared the presentation with him, and I will present the topic about the remote sensing for mapping macro litter on beaches and dunes. So when we talk about the beach litter survey by drones, we can draw up a framework that is composed by four stages. So the first two stages regards the planning field experience and drone field deployment. The third stage is the image processing and analysis. And the fourth stage is the litter survey assessment. So talking about this emerging technique, we have found in literature 15 works that use drone with RGB cameras to survey shores, both on Iceland and continental shores, but also lake shores. Most of these works were conducted in Europe, but also on the Arabic Sea and Maldives and Japan and China. And some of them, you can see the green dots, also adopted the drone to monitor the dune compartment. So we can also visualize better the framework. So what we did was like, we flew with drone both on beaches and dunes to map the macro litter items. So the drones collect images that can be processed to produce the orthophoto and the digital surface model of the beach and dune. The images can be screened manually or with automated techniques to detect the items and produce the marine litter maps on the area. In Duas Full Litter project, we also integrated the environmental forcing, namely waves, tide, and wind, and the beach morphology to try to understand marine litter pathways and dynamics. So I show you here a couple of pictures of the study site. So the first study site was the urbanized beach dune system of Lei Rosa, that is southern the Mondego River. We are at the central coast of Portugal, facing the Atlantic Ocean. Behind this promontory, we had also a natural beach dune system that is Quiaios, in which we tested the drone to monitor the dune compartment that is natural beach dune system in which the dune height reaches about 12 meters on the sea level. And as you can see, we are on a mesotidal coast with high energy. So the mean wave significant height is about 2 meters, but storms can reach about 5-6 meter wave height. Getting inside the framework, so as said, the first stage and the second one regards the planning and the field experience. So before checking the regulation, the local or national regulation for flying the drone, it is necessary to fly during a rainy and windy days. And it was convenient also to check the topography of the study area, especially to see if the presence of obstacles. This because during the flight planning that can be done in the dedicated app, it is necessary to set up the flight height. The flight height is fundamental because it determines the ground sampling distance, that is the image resolution. So as a higher fly the drone take pictures, lower will be the image resolution, as you can see here. Therefore, we found that a good GSD was about 1 centimeter per pixel. In literature, researchers fly between 5 meters and 4 meter high. This is a choice that can be done in the field and depending on what is the details of the survey that it is pretended. Overall, the battery autonomy is limited, so we have found that the maximum area that you can monitor is about 100 meter cross shore and 5 meter long shore. Here you have an example of 1 centimeter pixel GSD image, orthophoto. So once you have produced the orthophoto and the digital surface model, you can see here how the items can be recognized on the drone images. So the first we tested the manual image screening. So we tested and we produced a tool for GIS environment in which the operator can mark the item and also can input the type of item, the material of item that the operator can recognize. We also tested several machine learning and automated techniques for the automatic detection of the items on the images, such as random forest, CNN. And also we tested both pixel and object-based. The last one was, in our opinion, the most feasible, because it requires image segmentation that also allows to automatically measure the size of the items on the images. And here you have an example on how the measure of the size can give you also information about the dimensional properties of the items that were found in this case on dunes. So as the last work, we also tested the multispectral camera and sees the materials as a different spectral response, especially in the near infrared and in the red edge bands. So we tested the spectral angle mapper classifier to automatically characterize the material of the little bulk that we found on the beaches. So the last stage is the litter survey assessment. So by drone, as you can see, you can characterize the beach little bulk in terms of number of items, type and material of the items. You can derive also the source of the items and analyze the size of the objects. What we proposed, it was also that this kind of maps production. So once the litters are detected on the orthophoto, through the grid generation, we can count the items on the grid. And in this case, I'm showing here like the map of abundance in which we can recognize some hotspot. And then we can also produce specific maps, such as material maps. In this example, there is the map of plastic, but also source related, such as fishing related items. And finally, we can also show the actual area occupied by the litter in this specific area. This because in our view, the drone litter survey can not only improve
Anybody here? Connection is lost.
Yes, I think it was the panelist that was speaking that lost the connection. Maybe Louisa can check, or the chair for the session.
Yeah, I'll just have a quick check, but I think he may have dropped out.
Yep. Let's wait a second. Maybe he can try join us again.
Yeah, so everyone, we're just going to give it a couple of minutes just to see if he can rejoin and finish up his talk. If not, then we will go to the questions and answer roundtable session.
Hi everyone, we are having some technical problems. The connection of the speaker is down. So we are gonna wait a few minutes so we can see if he can come back to our meeting. So if it's not possible, we will start the roundtable, okay? So if you could just wait a few minutes. Thank you.
Meanwhile, if anyone has any questions about any of the last three talks from João, Filipa, and Umberto, please could you post them in the question and answer section, and I'll bring them up in the roundtable. Okay, I just got a message through saying that we think he's lost complete connection. So I think we will first off thank our speakers, João and Filipa, for their amazing talks. And we will start with the roundtable discussion. So we've had several interesting questions, and I've probably got a few to add there as well. There's a really good one that was, I guess it kind of came in fairly early, and I guess probably Felipe is the best one to answer this one. It's from Dina, and she's saying, how can one study microplastics in the diet of a top predator? What methods can one use? She's been studying the Cape fur seals by collecting scats, and she's just wondering, how would you recommend would be the best way to analyze that for microplastics?
Yeah, sure. Thank you for the question. And yeah, as I mentioned it, when we are not able, or the animals are iconic species or endangered species, we need to find a proxy of ingestion, and a way of doing that, it could be the feces, the scats of the animals, or for instance, on seabirds, the regurgitates as well. And there you can figure out at least a proxy of these ingestions. So I think that it's possible and it's advisable as well, because we, of course, don't want to injure some species. And of course, we wanted at the end to protect the fauna from the natural environment.
If I can add one thing, you can also go and search the diet themselves before the regurgitate. You know, for seabirds, there are some examples of that if you target the species that your target species is eating, you can have like an idea of what is happening. Yeah.
Yeah, particularly if you compare that with scat data, that would be useful. You can get an idea of the relationship between those two. There's a next question. This one, it says it's for João, but it's open to anyone here or on the panel. Do the data on the presence of microplastics in the coastal marine environment come only from scientific research, or is there kind of a government monitoring or NGO monitoring program for microplastics in the coastal marine environment? And they specifically mentioned in any European country, but I guess that any country, I guess, would be useful.
Well, when it comes to long-term monitoring data, it depends on what kind of infrastructure the country has. There are some countries who have like long historical data, who have been collecting it even sometimes with other purposes, and now we are using those kind of monitoring samples to go back and see if there was already like some traces, for instance, in our case, of plastic pollution. This is quite common in the UK, for instance, because they have like long series of data for plankton, and there are many groups in the UK that actually go back into this historical samples and do this. In my personal experience, I did the same when I was in Portugal. The first work that I kind of did assessing like zooplankton, we used historical samples that our institution IPMA had collected, and we traced back those samples. We try to focus more on macroliter other than the fibers, because we knew that those samples had been already processed, so we didn't want it to have a bias for cloth from fibers or fibers from clothes. And then I think Philippa pointed it out already when she shared like this this platform that was organized by the Alfred Wegener Institute in Germany, the litter base that Mina Tekman from Turkey developed, and her colleagues in Germany. It's quite comprehensive if we have like a global map of what is happening and what are the concentrations, because sometimes, you know, we can use that as historical data to see if baselines are changing. Then it depends a lot whether we are assessing sediment, or are we assessing surface waters, and are we assessing biota, because most of the times these baseline studies are one snapshot in time, so it's like a photo frame of that time and period for that region, and you need to have consistent data so that you understand the trends, so that you understand whether it is increasing, whether it is decreasing. And recently in a work that we did here in the ATU, we asked that question to experts, and depending on where they were working, they were saying, oh, it is decreasing for seabirds, it is increasing for beaches. So we also need to put it always in context of what is the matrix. But maybe Philippa wants to add something to this as well.
Well, in Portugal, we have, at least we are covered by the OSPAR Commission, which is the Northeast Atlantic area, and we have historical data or historical collection experiences, but mainly on beach litter. Microplastics is recent, and to my knowledge, in Portugal, we don't have yet a structure that covers microplastic monitoring in the entire country. What we have at the moment is contributions from the scientific community that feeds these national databases, and then integrates those databases in this OSPAR Commission database at the end. But still, as João mentioned, and I think that we should ask for more platforms, or at least an integrated way, because we are producing a lot of data, and we need to. This is the moment for data sharing and move on regulations, on policies, and we need urgently to cover the data gaps as well. And I think that monitoring is the best way of understanding the situation and the future as well.
Excellent. There's another question, and I guess it was aimed specifically at Philippa, but it could be for anyone on the panel. Do you agree that monitoring and measuring bioaccumulation of microplastics in organs such as the liver, stomach, and human kidney should be better disseminated by scientists?
Well, I would say liver from, it's not from human, right? It's just from the species, aquatic species, maybe. Well, of course, that when we are evaluating, I will answer for the aquatic species, because I think it's the way to know. When we target the gastrointestinal tracts of animals to infer microplastic ingestion, we know that most of these particles, could be expelled as well from the animals. And the transfer to organs is something important, but mainly for the lower particles. We are talking mainly about nanoplastics, because it's not that certain that microplastic can migrate from cells. And still the methods for monitoring those particles in these organs are still very underdeveloped, I would say, or not that easy as microplastic research area. But of course, it's an area of interest, because it will be related with the health of the animal and the performance as well, and the fitness. So yeah, it's important to do that. I think that we are not yet in the moment to monitor microplastics in organs rather than the gastrointestinal tracts or other proxies, for my knowledge.
And if I can add something to that, because I think that actually the question was really targeted to human health. Right now, we don't have enough knowledge or enough evidence, or even data to start making scientific-based approaches to say what is happening is this because of this. We don't have not a bigger number of samples across the globe. It's a very, very relatively recent field of research, and therefore it falls into the medical research. So it makes no sense to ask marine biologists or engineers, like my case, to be kind of describing what kind of like medical processes are going through when the human body is quite complex. So I would leave that to those experts, and I would wait until we have more data. What we can do, like Felipe was just saying, is we can continue to assess and monitor what we know on the marine species. We know, for instance, that plastic can absorb or adsorb pollutants at the surface and trace metals, as well having biofilm or eco-corona formations, and that might even contribute to have higher trace metal levels or persistent organic pollutants. And we have techniques to assess those on the species that we are targeting, the animal species that we are targeting. I don't know where we are on research on humans, and I would not be, Felipe as well, we would not be the experts on this. So that question should be left for the medical researchers or the people who do biomedical and those kind of research on those fields.
That leads me to another quick question. I did quite a large review of microplastics last year, I think, and the work that we kind of found was that the bulk of the studies are on amount of plastics that are in the environment, amount of plastics that are in the biota, as well as the type of plastics that are in there. But there were very, very few studies that were on direct impacts or biological impacts of microplastics on the biota. The bulk of it was, yes, there are heavy metals that are associated with biofilms, but not actually any behavioral changes or any direct biological impacts on many of the biota. So I just wondered if you could speak a little bit to that, either Felipe or João. I don't know if any of
do you
your
want to
groups are looking
do you want
at,
to go
looking at that.
first, or should I go first?
Well, I can try.
Okay.
It's not an easy question, of course, that we know that there are many laboratory studies that analyze different endpoints of effects of microplastics. And indeed, they are, well, thousands maybe of studies that related behavior, physiological, to fish, zooplankton, well, a lot of species are covered. I think that we are still struggling with what are the endpoints that we need to focus on. And of course, that in nature, microplastics are not the only contaminant. So how to exclude the effects of the other contaminants and the synergies between microplastics and the other contaminants as well. So I think that is not an easy task to detect the effect per se of the microplastics and the monomers that they are as well in the environment. So I don't know how to do that, to be honest. But I think that, well, laboratory conditions should continue using at least the levels of the environmental concentration that we found in nature. And we know that in that levels, it sounds that there is no significant effect in some parts of the world for some species, but still the levels are still increasing. So what is environmental concentrations of microplastics today? And so I think I'm not an ecotoxicologist as well, so it's not easy to give you a clear answer, but I think there is a cocktail of things happening in the ocean well, certain there is a lot to do in that research area as well.
And if I can add something.
Yeah, well, sure.
Thanks, Raymond, for this question. I feel like I have a different perspective on it, because from what I've seen published in peer-reviewed research or even in white papers, I've seen some effects and some impacts of plastic pollution. It depends on the size and on the scale, of course. You can see socioeconomic effects like loss of tourism revenue or loss of days at sea because of entanglement of the fishing vessels. When it comes more to species, like the question was asked to Felipe, there are few studies, but there are studies that show bioaccumulation in birds from one generation to the other on studies that have been conducted in Asia. Quite comprehensive studies where all the pollutants have been assessed, and where it clearly shows that the bioaccumulation is happening. Now this requires more data. This requires to be validated elsewhere. It then depends, going back to what Felipe was saying, on what kind of level do we want to see. Do we want to see cellular impacts, or do we want to see population or community impacts? Because they are different. And they require different tools to assess. I know of colleagues in Canada who are very, very interested in that question, and for years they have been compiling every impact that they have found from cellular level to community level, and they are really. I hope that some of it will come out soon. They sometimes show this process or this ongoing process in conferences, and we can see biological, physiological impacts of interaction with this pollutant through ingestion, like abrasion of the cellular tissue on the digestive gut, ulcers on some of the species. There are certain things that we are able more or less to assess or potentially relate to those kind of things, particularly when we are talking about sharp objects and that you are finding in species that you wouldn't necessarily find. This is very clear for seabirds that leave the nest for the first time. So some of them which might be like photosensitive, like the case of core issue water in the Azores. The majority of the birds who die before going out to sea and being able to live a normal life have been found with plastic inside their stomach, and this was the first time that they left the nest. So
yeah, I think the question was more on microplastics, but the other question was what's the best methodology for detecting microplastics in macroalgae? That's the last question, and I think we're probably need to.
Depends on the species that you are targeting, because macroalgae are, well, quite diverse, and the compounds as well to digest. I would suggest to go to follow the protocols that we share with you, because simple digestion, maybe, I'm not pretty sure if it works. It depends on the preservation as well of the species and the species per se. If it's more calcareous, maybe you will need more aggressive digestion. It's quite complicated to give you a clear answer on a simple protocol for a macroalgae for a group. But I would say testing first, something between the protocols that we share, me and João, from the water and the biota, and you will find out if it's visible or not. Yeah.
There are very few studies on plants that you could search. You could search on seagrass meadows, both laboratory and in situ experiments, and maybe take their approach, because it's quite different. You know, we can start with what Filipa suggests in her protocol, but plants have chlorophyll. We don't know if they react or interact the same kind of way. But it's a good question. Thanks for that, whoever asked it.
So we do have some additional questions, but I think we've overrun by almost 20 minutes now. So would it be okay? I think we mentioned before that it would be possible to respond to the additional questions either in the Answer Live question and answer chat or via email, if that's okay. And I will now pass back to Louisa.
Thanks to everybody for their contribution to this important discussion. The questions that weren't answered here will be answered via email, as Dr. Raymond Ward said. So here we finished this first day of lectures, and I would like to invite you to our side event, where students will present their research on human impacts and on marine ecosystems. You can watch it by clicking on the link that is available at the chat. Thanks to everybody. And a reminder for the students who will be attending to the students training, you need to get to LABOMAR to get your credentials at 1 p.m. until 1:30 in Brazil time. Furthermore, we see each other again tomorrow at 11:30 UTC time to our second day of event with the theme Oil Spill Investigation and Impacts in the Marine Ecosystems. So that's it for today. Thank you.